So welcome everyone to this educational symposium. Thank you for coming. My name's Sean Hatton and I'm just going to start off with some housekeeping. It's the left clicker, not the right. So we're going to have some interactive content in this presentation for the next 3.5 hours. So if you go to this website on your device, you'll be able to find the afternoon course, Researcher's Guide to ABCD. And in that, you'll see a social Q&A. So we're going to get any of your questions. We don't have time after each talk to do questions. So if you can just put it in here and upvote questions that you think are useful, and then the panel discussion will answer the top questions. Also, we're doing some polls, and they will pop up on your device as the questions come up. And so this is the first question. If you guys have it open, and you'll be able to answer that. Exactly how much experience do you have with ABCD, just so that we can pitch the talk to the experience level we have here. Oh, and it's not working. The Italians. Sorry. So we will be able to log these on the application, and what now? Hey! So, wow, a lot of you are new to ABCD, so that's interesting. All right, great. And with that, I hope you enjoyed this course and you get a lot out of it, and I'll pass it on to Hugh now. Oh. It's kind of locked now.
So left click?
Yeah, it's the left button.
Okay.
I can't get out of it now. Are you a technician? No?
Hang on.
Oh, okay.
Oh, he's over there. Yeah.
Okay.
So we're going to go back.
Is that your...
No, back.
Hey.
Keep going. Jesus. There we go. Okay. Hello, everybody. I want to start off by apologizing for my colleague making a derogatory comment about the Italians within the first 30 seconds of his presentation. And you think the slides are going to work for the rest of the day? Jeez. Okay. Thank you for coming. So my name is Hugh Garrivan, and I have now 16 minutes left. We're delighted that you're here. I mean, I think everybody in the ABCD consortium who is busy collecting data and working with the data, we're all, entirely 100% committed that we feel like our job is to try to collect and curate and analyze the best data set we can for everybody else to use. Like, that's our primary motivation, is to make these data available to you. So having sessions like this where we get to describe the data and we can answer your questions so that we can encourage and facilitate you doing the analysis is exactly our primary mission. And if we have to do it in Rome, so be it. Okay, so it sounds like quite a number of you are relatively new to the ABCD study. So my job here for the first 10 or so minutes is just to give you a quick overview of the study, and then we get into the details as the afternoon session goes along. So a few kind of punchlines. So there's the website. A lot of the information is up there. We have completed recruitment of just short of 12,000 kids. So this includes, by design, over 2,000 twins, half of them identical twins. We have 30 triplets by accident. We have no idea what the research question is, but somebody really should do something with the 30 triplets. And all the kids are aged 9 and 10. The plan is at 10 years of assessments, so we have interactions with them about every 6 months. But Terry is going to talk through the details of the assessment procedures. There's extensive neuroimaging. You're going to hear about a lot of that in today's sessions. Genotyping, lots of psych assessments. Again, Terry will cover a lot of that. Hormone analysis, geocoding. So we have the facility that if you have any geolocation information that you would like to try to merge with our data, that we're going to try to facilitate that. And also the plan is that what's appropriate for these kids at age 9 and 10 is likely going to change. So we anticipate changes to the protocol and to the assessments as the kids age. And then the critical thing, and again, in part, no doubt, this is why you're here, is that these data are publicly available. So every year we have a fully curated annual data release. And again, our goal is for you all to analyze it and to have as much fun with it as we have collecting it. Actually, more fun with it than we have collecting it. But okay, a few just kind of backgrounds. I'm sure because you're in the room, you probably know this, but why focusing on adolescence? As you know, I mean, lots of interesting things happen during the adolescent period. Kids between the ages of 10 and 20 change profoundly. And also, this is the period where a lot of mental illnesses show their first earliest signs. And then also, neuroimaging has revealed, we have substantial brain changes going on in this period. So it's a critical window of time, we think, and really trying to understand it is, it is important. We know, from lots and lots of literature that genetic factors, environmental factors, behavioral factors are all associated with, good mental health, good health, outcomes. What we don't know, though, I think, is how all those things interact, right? I mean, much of the adolescent research is cross-sectional, is with small samples, where one might study one of these factors, but trying to put it all together. So we know that nutrition is important, we know that sleep is important, we know that exercise is important, but do we have good quantification for it? Do we have a good understanding of how, doing well in one domain might counteract, negative impacts of the others? So, that's the motivation for this study. And that's why our sample size is so large and our assessment battery is so broad. Okay, so just by way of where this came from, so this is coming from the Collaborative Research on Addiction initially from out of the NIH. So the impetus, the starting point for this study was substance use. And I think even more particularly in the US and across the world, with cannabis becoming decriminalized or legalized, there were concerns about, do we really know what does cannabis do to the developing brain? And we really need to, if we want to understand that, maybe a large longitudinal study. So that's, I think, where it kind of initially started. But then, of course, as the study got bigger, you might imagine then other institutes came on board. So, of course, mental health says, well, if you're going to be doing this large longitudinal study, can we add the following assessments? And that's how the project got bigger and bigger and has for sure moved beyond just a particular focus on substance use. And all these institutes have come on board. We have a lot of federal collaborators. The data are collected across 21 sites in the US. So a pretty good geographical spread. These are the institutions that are involved. The nucleus, the hub of the whole project is at UC San Diego. So that's where we have the coordinating center that coordinates all the 21 sites, as well as all the data analysis and informatics happen there. Okay. Study objectives. Again, much of this will probably, I probably don't need to belabor this, you probably, are aware. But, to try to articulate it, the first kind of primary goal was to develop national standards for normal brain development. So, we have height and growth charts for kids. We don't have anything comparable for brain, right? So what does typical neurodevelopment look like? What do different developmental trajectories look like? What are the factors that can affect these? Again, as I mentioned, there's a lot of research, but how all these different factors might interact with one another and how they might affect different subsets of the population. These are the sort of questions that we want to address. How do genetic environmental factors and their interactions affect development? We know, as I mentioned earlier, health, physical activity, sleep, bangs on the head, all these things are potentially important, but how do they combine? How do we quantify the impact that they have? And of course, mental disorders is going to be a critical issue. Again, with a longitudinal study like this, just to be super duper clear, we won't be able to identify causal mechanisms. This is still a non-intervention, longitudinal assessment, but certainly by having rich assessments at age 10, and then seeing how those kids develop, and seeing what sort of outcomes might be predicted by the baseline assessments, certainly helps to narrow down the focus of what are probable causal mechanisms. And then finally, determine how exposure to various levels and patterns of alcohol, nicotine, cannabis, caffeine, and other substances might affect development. Again, same issue. We can't really ascertain causal mechanisms, but if we have baseline, similarities that then following, let's say, years of substance use, we see group differences, again, it helps identify probable consequences. Okay, one question we get asked, and I thought I might just focus on this a little bit, is why is the sample so large? 12,000 is a lot, and that's a lot of money and a lot of resources. And people might ask, is that too much? So here are just some of my thoughts as to why 12,000 is, actually no sample is big enough. It could always get bigger. The first one is a gimme, right? If we can't come up with a comprehensive survey of neurodevelopment, then we've not done our job correctly. But with a large sample like this, it allows you to study specific groups of interest. So if your particular interest, and it would be kind of curious to survey the room, but let's say it's depression, or childhood anxiety, adolescent anxiety. You don't know in advance who the kids who are going to develop that adolescent anxiety are going to be, right? I mean, you could try to get an enriched high-risk group, but then you have this sort of potential ascertainment bias in your data. We don't know what the trajectories of these kids will be. So over the coming years, a substantial number will, develop psychosis or depression or eating disorders or substance use problems, or will do terrifically well. Will academically excel, will become expert musicians. Who knows what. We don't know who they are in advance. So the idea is to have a very large sample and then follow them, and then the people will will categorize themselves into the groups that they're going to become, and then we can go back and assess. And another issue that people often don't think about with the large sample size is, as you well know, for most of our cross-sectional comparisons, if any of you have ever tried to publish a cross-sectional paper, there's always concerns about confounding variables. Well, did you control for? Do your groups differ on? If you have a group of interest of just 100 kids, you now have the opportunity to search amongst the remainder of the data set for kids who are matched on everything but the particular variable of interest. And being able to do that is actually hugely, hugely valuable. This really gives us a chance to be able to control for all those confounding factors. We will have sufficient statistical power to detect small effects. You're going to hear a lot more about this as the afternoon goes on, but I think we are of the consensus, if I can say that most effects that are in the literature that are true are small effects. And if you really want to detect small effects, you need large samples. We can do rigorous data analysis. So we can, you all know within neuroimaging there's this reproducibility angst. We can build in appropriate cross-validation and replications if you have a large sample size, and we'll talk about that later. If we have sufficient individual variation, then we can disentangle demographic factors that are very often confounded. So things like, for example, race, urbanicity, socioeconomic status. If we have enough people, we might be able to tease apart these effects that very, very, very often co-vary. And then also, we can see if effects vary with subpopulations. I think everybody is aware that far too much research is done on white males. And so this gives us the opportunity to stratify our sample to see if what might be a causal mechanism might actually differ depending on demographic factors and so on and so forth. Okay. And just on that point, this kind of speaks into this move towards population neuroscience. Our goal in this study has been, actually quite different to most studies. Most studies, if you're trying to study, some phenomena, you want to try to control for all these other confounding variables. Our approach is actually the opposite. Try to have as few exclusion criteria as possible so that we can kind of capture all that heterogeneity in the population. And then to try to use, sort of epidemiological practices for recruiting the sample. And this would be a good time to mention the representativeness, would it? Yeah, why not? We do want to make this point because this is important. We have made big efforts for the sample to match demographically to our target population, which is the US population of 9 and 10-year-olds. And as you'll see in the coming slides, we think we've done a pretty good job of it. It's not perfect, but it's pretty good. But we are very cautious that people not think of this as therefore being a representative sample, right? I mean, we have 21 sites. We didn't randomly select those sites. Not everybody in the entire population in the US had an equal opportunity for participating in the study. So it can never be truly epidemiologically, rigorous. It's not a true representative population, but it's pretty close. And we went to lengths to try to ensure that we had very broad representation of a whole host of demographic factors in the final sample. And that's where I'll talk you through next. Okay, so just a few slides now to tell you about what the cohort is looking like, and then we'll take a few questions. So for people in the consortium, this will probably induce flashbacks. We had a very aggressive recruitment window. So we started in September, October of 2017, and we gave ourselves 2 years across the 21 sites to recruit eventually about 12,000 kids. So recruitment just came to an end in October of last year. So this means we have baseline assessments, and you'll hear about this on everybody. We have 6 months assessments on everybody, 1 year assessments on at least half, and so on and so forth. And all our sites worked extraordinarily hard to reach their recruitment targets. Most of the recruitment was done through schools, so this is a bit busy. The twins, as I mentioned, because twins are rarer and probably wouldn't be very efficient to try to recruit them through schools, they were recruited from twin registries. But the remainder of the samples, or the general population, were recruited primarily through the school system, because 98-99% of all kids have some contact with either public, private, or charter schools. And not to go off into this in too much detail, but if anybody wants to, we'll happily elaborate more. We had a very careful sampling of the schools that fell within our site's catchment areas so that we could choose them so that we would have this broad representation of sociodemographics. So mostly the kids came from schools, but we had a few other systems. We allowed people to refer we allowed participants to refer friends, thinking that would help keep the retention. We started off by having a few mailing lists just to get kind of kickstart recruitment. We found as the study went on that we really needed to make extra efforts, especially to recruit lower income and minority families. So we did much more outreach into those communities to ensure that we increased numbers there. The sample, this is, we're used to looking at it broken down by our sites, but we have about 52%, sorry, just to orient you, does this work? Maybe. So here's the ABCD average, and then these are just our individual sites. I think it's 52% male, and 52% of the sample were 9 and 48% 10. So, and all very strictly between 9 and just shy of 11 years of age. Demographics. So to orient you here, what we did is we set targets based on the American Community Survey. So this is a survey of 300,000 to 400,000 families with 9 and 10-year-olds in the US. So those were like our target populations, and they're represented by the inner circles. And then the outer circle are the demographics of the sample that we recruited. So if you can see there, to orient you for race, for white, so the inner circle, 49% of the US population of 9 and 10-year-olds are white or Caucasians, and our sample contains 50%. For African American or black, the sample in the population in the US is about 13% black. We aimed for 17% and recruited 15%. And then you can sort of follow the circle around. So we have, again, pretty good representation across races and ethnicities. We've broken it down by the general population and also by the twins population. So again, is it perfect? No. But we have broad representation across all these demographic groups. Similarly, for socioeconomic status, again, to orient you, I can't look, I don't have eyes in the back of my head, so I don't know if you can see my mouse. Yes, you can, great. So here's the American Community Survey, so these are our targets. Here's our twin sample, here's our general population, and then it's just broken down by the highest education level in the family. So you'll see, just by taking a quick moment to look at that, two things. One, our sample isn't perfect. As you might imagine, it skews slightly towards better educated families, and it skews slightly towards families with higher incomes. And I think that's probably just the nature of these sorts of studies as to who you can, who's interested to come and participate. So the matching isn't perfect, but what we would like to stress is within our sample, we have broad representation in all these groups. And again, this is the beauty of having 12,000 participants. So, a relatively small percentage still translates into a lot of kids. And then one can get even more fine-grained with the breakdown. We can now look at family income as a function of race and ethnicity. So of course, as you get into those finer and finer cells, then any kind of imperfections become a bit more apparent. On a number of other factors, I won't belabor this either, but not by design, but just making sure that things like family size, the percentage in the labor force, we have again pretty good matches to the population. Again, I think I know this is I'm spending a lot of time on this, but for the conclusions that one might want to make with this nationally important study that's got a very broad representation, we think it's important to be aware of the demographics and get a sense of how well they do indeed fit. If we want to make claims about this is what typical adolescent development looks like, we want to know that we're capturing the heterogeneity in the population. And then I'm not going to talk about this. If you want more details, Wes can talk you through it. But in the data analysis machinery that Wes will be describing, there are ways that you can actually adjust your sample to map even better to population demographic percentages and ratios. Okay, so sort of propensity weighting procedures that are already built into the systems that Wes will be describing later. Okay. Last couple of slides, 1 minute, 30 seconds. How's the study going? So far, it's going really well, right? Fingers crossed. We have a very motivated, very interested sample. Our sites have really gone to an awful lot of trouble to make the experience as much fun as possible. There's a huge amount of creativity, making the day of assessments kind of fun for the kids. They accumulate points, they win prizes. We have all sorts of outings and events like softball games that we bring them to. They get t-shirts, they get pictures of their brain. We can quantify this. So we are, like I say, about 2.5 years into the study. Our absolute number of kids who have withdrawn from the study is really, really small. So we started off with 11,800. Here it says 75, it should be 78. We've had 75 withdraw so far. So this is great. Will this last? Yeah, probably not. But we're going to do our best to try to keep the sample as solid as we can. The thing that we're really focusing on at the moment then is not so much kids who are withdrawing, but kids who are missing assessments. The idea being that if you miss a 6-month phone call or a 1-year visit, then there's an increased probability that you might withdraw from the study. And we are already seeing that, surprise, surprise, missing assessments are not evenly distributed amongst the sample. Right? Again, it's the lower income families that are finding it most difficult to make, our scheduled appointments. So, we're on top of this, and our job for the next 10 years is to manage this and to ensure that we can minimize the selective attrition as much as possible. And finally, if you want information, so like I say, I'll just point you at the website. Our goal is to hopefully try to keep as comprehensive a list as we can of the publications that are out there. This is an open science model. We have no control, nor do we want control over what people choose to do with these data. You articulate your hypothesis, you grab the data, you do what you wish with it, and what we will try to do is just try to stay on top of it so you can see, and we can see, what the field is doing with this data set. Are there any? To the end of everything. So not even quick questions. I'm fine. Get up.
What button? on the mouse. Okay, right. Okay, so I'm Terry Jernigan, and I'm a consortium coordinator for ABCD, and I'm going to be telling you more about the assessment protocols. Full disclosure, the person in our leadership group at the coordinating center who knows everything there is to know about the assessment protocols is Susan Tapert, Professor Susan Tapert, and she couldn't be here, so I'm going to be filling in for her. So try to be kind to me. I'm going to cover a lot of territory, and it's a very complicated protocol, and I will certainly try to give you as much information as I can, and we can answer questions later if I can't answer them all today. But I have a few slides that I'm going to go through kind of very quickly because they're very much overlapping with Hugh's, because I wasn't entirely sure what Hugh was going to say because he wasn't available. He was in Tuscany. But anyway, so we'll go through some of those quickly. But I just wanted to, let's see. You said right? This goes, oh, left, okay, good. All right, so you just heard about this, so I'm really not going to go through it. I was going to start by telling you about the rationale for ABCD, just like Hugh did. Just is because it's really the rationale for the protocol as well, of course, but you've already heard about this, so I'm just going to go through it. Excuse me. But I am going to I think I have one sort of objective slide. You've heard about how many, so this is an important point that I would have made at this point if I'd been presenting that, was to say that as Hugh mentioned, this study was initially motivated primarily by the addiction institutes and by NIDA in particular, and National Institute on Drug Abuse, and there was a strong interest in understanding about substance use and substance exposure and risk phenotypes for substance use. But as the number of additional federal partners grew, then obviously the objectives grew a lot. And people wanted us to answer a lot of other questions that were not central to the substance use questions. And this is in part what has really driven the decisions about what's in the protocol, is trying to answer all those diverse questions that came to us from the additional sponsors that have gotten interested in the potential for AB to answer questions with ABCD. Okay. And this is the same slide as Hugh's, but I'm just emphasizing in bold a few things. We were supposed to actually, understand a great deal more about normal brain development. You're going to hear more about that when we hear more about the imaging data. But we had, this goal of measuring these individual developmental trajectories across all those domains that you see there: brain, cognitive, emotional development, academic progress, mental health, lots of different things. And of course, we have this embedded twin cohort within there. So we have a lot of questions about genetics as well as genomics. You'll hear about that. But because some of the other institutes that are interested in physical health, we are now looking at physical health, physical activity, sleep. And of course, because of the size of the cohort, we will be observing the onset and the early progression of certain mental disorders. So we will see some participants develop these disorders. And we want to really try to understand those better, what's influencing the course or severity of them. And then back to this original important goal that we had to understand exposure, substance use exposure on the developing brain, and also, what kinds of risk phenotypes might be present early before substance exposure. And so that's very much why we're recruiting at 9 and 10 years of age, is because we have some hope of seeing kids before they have any substance use exposure at that point. Okay, here's the basic visit schedule. There are these comprehensive assessments at baseline and biennially thereafter, which include all the multimodal imaging and all of the domains. And then we see the kids much more briefly on interim years, just to kind of check in with them and do a shorter battery of things. This is in part just to keep connected to them. And then we have even briefer 6-month phone web kind of assessments that we're doing. And I'll tell you a little bit more about those. So this is kind of an orienting slide. I'm going to tell you, I have a lot of slides that I borrowed from Susan, and I'm going to go through a lot of details, at least to expose you to the detail of the protocol. But this is just to point out that the protocol involves both a youth protocol in which we're collecting data directly from the youth themselves, or measuring things directly in the youth themselves. And this is this is kind of the basic structure of the domains that we have focused on in ABCD. This is for the baseline assessment. It happens to be for the baseline assessment. And you can see that the domains include physical and mental health, neurocognition, what we call culture and environment. And again, I'm going to dig down under these in a minute. Substance use, of course, very important to us. Brain imaging, you're going to hear a lot more about from other people, but you can see right there the basic structure of this. I don't know if you can see this, yes, you can see that, right. You can see that there's the usual structural modalities, but also there's resting state, and then there are 3 task fMRI paradigms, and I'm sure you'll hear more about those. And then we have some biospecimens, and they're all there for a particular reason, and I'll kind of go through them for you and tell you what we're collecting so far. And then we have these other data sources, and Hugh alluded to this, that we have geocoding, and so we've been able to collect some data from public databases and add some sort of environmental and exposure variables relevant to the kids' neighborhoods and their exposures. Also, we're trying to collect other, what we call passive data, things that we can collect, say, from their schools, or hopefully eventually from their medical records, and from their teachers in some cases. And so we don't assess them directly, but we get additional information about them. Okay. And of course, at 9 and 10, we have to collect a lot of information from their parents. And these are the main domains for the assessments that we do with the parents: physical and mental health, substance use, and culture and environment. Okay, now I'm going to go into the details. And I know this is almost impossible. It's even impossible for me to remember how to describe it. But there is a very detailed and carefully designed protocol for trying to detect, in some sense, the initiation of substance use in these kids. And there we have an excellent group of investigators this has really been their focus of research for their whole careers, and they're very good at this, and it's very carefully thought through, because it's a difficult thing to do in a developmentally appropriate way in 9 and 10-year-old children. It's very important that you ask questions in the right way, and that their parents do not object to the kinds of questions that you're asking them, and things like that. So it's been very carefully designed so that we really begin with 9 and 10-year-old kids, and this is the youth protocol I'm talking about here, by finding out what substances the kids have even ever heard of. So that's kind of how the assessment begins. And then follow-on questions only occur if a child has heard of a substance. And then if they've heard of something, the next set of questions are kind of, well, have you had a sip at, if it's alcohol, have you had a sip at a wedding or something like that? So you kind of move into the assessment to figure out whether there has been the initiation of any use, and then if there has been, what's the regularity or the frequency of that use. And then, of course, a lot of other assessments get triggered. But until there's some reported use, very little is asked further. And so this is kind of going to unfold over time. In the 9 and 10-year-old kids, we were very pleased to find that they were essentially naive. There were extremely few instances where kids had begun to use any of the substances that we asked them about. And that was really reassuring, because we wanted this to be a naive baseline for this set of aims about substance exposure. So, but we are already in our, subsequent follow-ups beginning to see a little uptick in some early exposure, experimentation, and so on. And so, and I think our protocol has been well designed by the people who have worked very hard on it to be developmentally appropriate and to roll out so that we detect the onset and that we get more and more detailed information about what the kids are being exposed to as we go forward. Now, most of this is, I'm focusing now on the part that's sort of self-report by the kids, but you'll see here at the top, this is, there are these sort of biospecimen assessments or assays of substance use. And so there's a percent, and there's this XX, because this is a little hard to say what percent that is because it's gotten a little bit complicated, but a small percent of the cohort, even at the baseline, were asked to do the breathalyzer, even though they were 9 and 10 and they were not reporting anything, just a small percent. And also some saliva for drug screening, and a Nicolert test in the urine for for evidence for tobacco use or nicotine, and so those are there to just make sure that we kind of look to see how often are we seeing something we did not expect. It's associated with kids not telling us things, or there being exposure that they're not reporting, and so on. It's a small percentage at this point. If once a child has begun to report that they have some use, then they're tested every visit subsequently with these biomarkers. And you'll see that we do collect hair samples for all the kids, at least all the ones who'll let us. And so that we do have hair samples that we're keeping. We haven't analyzed many of them. We do have a subset that have been analyzed so far, just again to get a baseline of what we're seeing. But they're being stored, and we will have the hair samples in the whole sample as we go forward every 2 years, I believe. Okay, so this is very complicated, and I realize it's hard to read, and if I tried to take you through it, I'd get it all wrong. But what the main point is that it's kind of a lot of skip logic here, where you get more information as you see more initiation of use, or more self-reported, and that's just the end of it there. But you'll recognize, if you do substance use research, you'll recognize a lot of these questionnaires and things. And there's a timeline follow-back procedure which is used once you begin to have use that's worth reporting. Okay. Next domain is neurocognition. Again, this is the youth protocol. This is the part where we measure things directly from the kids, and you'll see that it starts off, as you can imagine, with some vision screening and something about hand dominance. And then we use the NIH cognition toolbox, and so I don't know how many of you guys have used that or have some experience with it, but it's a fairly brief neuropsychological test battery, kind of, iPad administered in our case. And so these are just the subtests here, vocabulary. There's a flanker, a pretty classical flanker task, a list sorting, a sort of working memory task, a dimensional change card sort. Sorry, that's misspelled. And the pattern comparison test, it's just like a processing speed test. There's a picture sequence memory test, which is kind of an episodic memory test, oral reading recognition. And then we have some additional tests that we've augmented the toolbox with. There's a list learning task, the Ray auditory verbal learning test, a matrix reasoning task. The little man task is a spatial problem solving and mental rotation kind of task. Cache choice, one trial, quick task. I'm sure most of you guys know about that. And then there's a delay discounting task and an emotional Stroop task. But you can see that these two tasks, the delay discounting and emotional Stroop, didn't make it into the baseline, but we added them on at the 1 year. So you can see that they would be there. And I guess, I don't know if anybody's really talked about this, but I think somebody mentioned that you'll have data in the 2.0 data release that includes essentially all the baseline data. And then whatever was available in the follow-ups that when we cut to do the data release. So we did try to time the data release so that we tried to get as much of the baseline data and all the baseline data that we could get there. But then you kind of just got whatever the follow-up data was available at that point. So you will see some 1-year follow-up data there, but not the entire cohort, of course. Okay, now mental health, again, the youth protocol. We have a prodromal psychosis questionnaire, a fairly new one, it's a really interesting new method. The brief problem monitor scale that many of you probably know about in the youth. A delinquency measure, life events questionnaire. I'm going to talk in more detail about the KSADS. The KSADS is a well-validated psychiatric interview for mental health symptoms for children. And I'll talk more about it. There are a couple of measures of impulsivity, sort of resiliency items. We do have a protocol throughout for looking at gender identity and sexual health. Again, a new domain that is being kind of delicately moved into the study. And a mania subtest and a few NIH Toolbox positive mood items here. And again, you can see that some of these things weren't done at baseline, but they were done at the follow-up, 1-year follow-up here. And then this is mostly information from the parent about the child's mental health, some about the family's mental health. There is one measure of the parent's own mental health, which is the first one, but then the rest of it is mostly things where it's parent report about the child, CBLC, the child behavior checklist, demographics, family history, again, more gender identity and sexual health, a general behavior questionnaire, life events, and this a short version of the social responsiveness scale. And again, I'll go with the case ads. Okay, and this is busy, very busy. The case ads has many modules. It's a diagnostic interview. I mean, you can find out all the information online about it. It's very well documented. But there are many modules. And what we're trying to do is, in the youth, we only administered at baseline the ones where you see ones, right? So we looked at mood disorders, social anxiety disorders. These are modules now. Sleep problems, suicidality. Those were the things we did in the kids at baseline. At the 1-year follow-up, we added a little bit more: alcohol use disorder, drug use disorder, and we repeated suicidality, as you can see there. For the parents, we did a lot more. So we collected a lot more information from the parents about the children's mental health at that point. So you can see that we did a lot more of the segments here because they're really more appropriate for parent at this age, parent report. So a lot more of the anxiety and obsessive compulsive disorders, eating disorders, oppositional defiant conduct disorders, tic disorders, and post-traumatic stress disorders. You can just see that there's a whole lot more of the modules that were included at baseline. And we added a few or repeated a few of those at the 1-year follow-up here. Okay, so here's the youth assessment for physical health. We're moving to physical health now. We do anthropometrics, of course. We took mostly saliva samples, almost all at the baseline. We got salival samples and sent those off and got DNA, and those have been genotyped. So there is now genotyping data available for most of the sample, and that's in the 2.0 data release. We also collect saliva samples annually, actually, for pubertal hormones. And then we do some pubertal development scales, and this again is with the youth risk behavior and physical activity, information about physical activity, but that's self-report. And then again, you can see that we repeat some of that stuff at the 1-year follow-up, including the saliva samples for puberty, for looking for hormones, and this scale, but we didn't repeat these two things. Okay, now we collect a lot more information about physical health from the parent. One thing we do is we ask parents if they'll give us baby teeth. So they are giving us shed teeth. Turns out there are a lot of interesting things you can learn from baby teeth about even fetal exposures in some cases and into postnatal life. And so we are trying to collect as many baby teeth as we can. We get some developmental history from the parents, a medical history. We learn about all the medications kids take. We don't get any nutrition at baseline, but we did add it on at 1 year. We get a traumatic brain injury scale, a pubertal development scale from the parent, sleep disturbance scale, and from the parents we get a lot of information at baseline about sports activities and other kinds of activities that the kids are involved in. Culture and environment. What do we mean by cultural environment? Well, you can see this is kind of a mixture of things. If you know some of these assessments, it does involve learning a lot about acculturation factors in our very, very diverse cohort. Things like, cultural and religious practices and things like that. There's also a lot of information here about family environment, family dynamics, family conflict, for example, parenting styles, or at least the perceived parenting from the child's point of view for this one. There's a little bit here for sort of stereotype threat kind of stuff, perceived discrimination that the kids are experiencing. Some pro-social behaviors, and a little bit about their school environment, and at least their perception of their school environments from this self-report battery. And again, we've added a few things and repeated a few things at the 1 year follow-up. From the parent, we get a similar kind of assessment from the parent's perspective. So again, information about the family's acculturation, the family's environment. Some of these things that say Mexican-American cultural values, for example, actually are very general. You get a lot of information about that are across, it has a name Mexican-American. It does cover a lot of very relevant dimensions for the Mexican-American population, but it's actually very general. You get a lot of interesting information about what the family is doing, what their community is like, and what their community involvement is like, and so on. And we get information about neighborhood safety and crime. I think those are kind of self-explanatory. Now, for mobile technology, what you see in the 2.0 is mainly the screen time questionnaire. So we get information about what is the self-reported screen time by the youth and by the parent. We are in the process of doing a Fitbit study, and so that next data release will probably have Fitbit data in there, and that will be added to this mobile technology domain, but it's not there yet in the data. And then just to finish off, we do this mid-year very brief phone assessment. You won't be surprised to hear that this is motivated a lot by trying to catch that first use of substances, which is very important to our lead institute. And so we're checking in with the kids about their substance use, and we're asking them a few questions about from the brief problem monitor scale. But then we put in these NIH toolbox positive affect measures, partly because we want to know, but also because we want to lighten it up a little bit. So we have a brief interview with them, and it's really to check in on a couple of these domains where we don't want to miss the timing of something happening or developing or showing up in their lives. Okay, so I know we're not supposed to take questions, but I'll stop there and pass the baton here. Thank you.
Hi everyone, Sean here again. Just quickly, this is what we're going through today. I just want to advise that this presentation has a lot of slides after the presentation. We had to throw out a lot, so when these slides are shared, there's a lot more information, about another 20 slides of information. And just as a reminder for people if they've come in late, if you have your smart device or anything web-enabled, if you go to this website, we are taking the social Q&A for any questions during this session, and some questions are already coming out now. And we're also doing some polls during this actual talk. So, on to Don.
Thanks. So I'm going to give an overview of the imaging that's being done and the processing pipeline for that. We have a description of the processing pipeline that's currently available on bioRxiv as a preprint. The publication is currently in review and hopefully be out soon. So the processing pipeline is sort of conceptually organized into these different stages of unpacking and conversion of the raw data to some pre-processing and then brain segmentation, then an analysis where we're extracting information from the imaging data, and then a summarization that is putting the data into a tabulated form that we can share and use for statistical analysis. This is a kind of complicated diagram showing some of the outputs of the processing pipeline. I won't go into all the details, but basically we have the data coming in, raw data being unpacked, and then some structural data going through FreeSurfer, and we have fMRI data, diffusion data, and different analysis outputs that are created. So the first big section of structural processing, we have the T1 weighted and T2 weighted images. Some of the processing steps are gradient distortion correction, registration between the two modalities, T2 and T1, and intensity non-uniformity correction, and then a resampling into a isotropic voxels in a custom atlas space, or rigid body registration. And then finally, the brain segmentation, cortical surface reconstruction using FreeSurfer. And at this point, we are only using the T1 weighted images, although in the future, we can use the T2 to get better segmentations. So for the analysis of the SMRI data, we are extracting different kinds of measures. The first is the morphometric measures like cortical thickness, area, volume, sulcal depth, and so on. Image intensity measures, where we're taking the T1 or T2 weighted images and then calculating averages for different regions of interest. We also calculate a cortical contrast measure that's the difference between gray and white matter. We do this for cortical surface ROIs using some of the standard FreeSurfer parcellations, and then subcortical ROIs to get intensity measures and volumes. For diffusion MRI, we have two different types of analyses. First, diffusion tensor imaging, that I'm sure everyone's familiar with in imaging, and restriction spectrum imaging, which is something developed in-house that is more of an advanced diffusion analysis method that takes advantage of multiple B values. And I'll explain a little bit more about both of those. But first, the diffusion processing includes head motion correction, correction of spatial distortions like B0 field inhomogeneity, eddy current distortion, gradient nonlinearity. There is a robust tensor estimation to identify dark slices that can be caused by abrupt head motion. And then there's a registration of the diffusion images to the T1 structural images. So for the measures derived from the diffusion data, we use DTI, like I said, to get things like the fractional anisotropy and the mean diffusivity. The restriction spectrum imaging gives us a way to separate the diffusion into restricted and hindered diffusion pools that are roughly equivalent to intracellular and extracellular signal fractions. And then for both of those types of measures, we calculate ROI averages. So for white matter tracts, subcortical structures, and the cortical parcellations. So there's resting state fMRI. The kids spend about 20 minutes just staring at a fixation cross. And the pre-processing for the resting state fMRI is the same as what we have for the task fMRI, and I'll get to more about the analysis of that in a bit. But so the fMRI pre-processing includes distortion correction for B0 distortions, gradient nonlinearity distortion, and then there's head motion correction, which uses sort of standard volume-wise registration. And then finally, there's a registration to T1 for being able to extract using the structural regions of interest. So for resting state fMRI, there's some additional pre-processing that includes removing the initial volumes and then filtering the data to normalize any trend, regress out things like motion and global signals. There's a censoring that's done to remove frames with too much motion, and finally bandpass filtering to look at the slow oscillations. And then the time series are resampled to the cortical surface, and so we get average time courses for different cortical regions of interest and for subcortical regions. And then we use a seed-based correlation analysis using this Gordon parcellation that is divided into different networks. And so we're able to calculate the pairwise correlations between ROIs and get a summary measure of the correlation within and between networks, and then between each network and subcortical ROIs. So for task fMRI, there are 3 different task paradigms: the monetary incentive delay, the SOP signal task, and the emotional end-back task. And in addition to getting the sort of brain activation for each of those tasks, we also have behavioral performance measures for each task. So a little bit more about the tasks themselves. For the monetary incentive delay, sort of depicted here, where the subjects are pressing a button, and there's a risk, they're winning money, and so they're kind of, it's designed to get at risk and reward. And some of the contrasts of interest are things like the anticipation of a reward versus no reward, or the feedback of a win versus the lack of a win. Or so then the stop signal task is also a button press in response to a cue, and there's a stop signal that is supposed to, the subject is supposed to withhold their response, so it's getting at inhibitory control. The emotional end-back task is like a regular end-back, except there are pictures of faces with different emotions, so there's sort of extra things that can be extracted from this, but the main thing is working memory. And so the analysis for the task fMRI that's specific to the task fMRI, also removing some of the initial TRs to get rid of the signal where the T1 decay. Then there's a GLM used using AFNI's 3D deconvolve. We include nuisance regressors, such as motion and their derivatives. There's censoring of time points with excessive motion, although with a bit more liberal threshold than what's used for the resting state. And then the hemodynamic response function is modeled as a gamma function with temporal derivatives. And then the GLM coefficients, and well, so this is not quite right. So we have the time series that are averaged for the different ROIs, and then we do the GLM on those ROI time series, and so we'll get a GLM coefficient and a T statistic, or actually standard error that we calculate from the T-stat. And so we have those beta weights and standard errors for the different subcortical regions and cortical parcellations. There is task fMRI behavior scores that are then calculated from the response times and accuracy, and so we also calculate a overall behavioral performance flag that can be used to exclude subjects with poor performance who weren't really paying attention, perhaps. And we have a number of different areas for future development, including longitudinal analyses. As we get the follow-up data, we'll be incorporating more specific longitudinal comparisons and image-based analyses. Sean, is this your part now?
Okay, so now I'll be talking about data sharing and some of the tools and recommendations that we have with the data. Oh, that's a silly button. So our data is shared with the NIMH Data Archive that originally was an autism research group and then expanded. We're one of the largest groups, along with the Human Connectome project, so we're working with them very closely for them to handle the large amount of data that we give them. The nomenclature for ENDA, the collection is a whole grant, the entire study. So whenever you hear study, the study is actually a release that we have So that's the ENDA naming conventions, and then they have data structures or instruments, which are associated tables with data in them. So yeah, the ABCD as a whole is collection number 2573, and at that stage we got to the right. Fast Track is a DICOM sharing service. It's not tied to any release, and the same with the genetic data. It's part of the collection instead of releases, which are the annual curated releases of the data that Don has just mentioned. So if you go to ENDA, this website, this is where you get it. There's some really good tutorials, but also they've put this little button up here to get straight to ABCD, which is nice. And you'll come across this page, which is usually all you need for it. Most commonly accessed data sets will bring you to the current release. And also this is to access deep for the first time, but Wes will talk about that more shortly. So Fast Track. Fast Track is raw DICOMs continually updated. We're working towards a kind of monthly pattern. At the beginning of the month, there'll be fresh DICOMs available. This is just, we're collecting them continually and updating them to ENDA continually, but ENDA has to go through some checking before it actually gets updated on the website, so that's why it's a little more sporadic than constant. Also, we're going to be including shortly instruments to advise of changes. We've had one subject whose ID was put in wrong at the site, so the actual DICOMs, that's the only time we've had a problem at that level, so one person out of 12,000. And we're also going to add in a QC score of a pass or fail at the raw QC, just so if you don't have to process files which are completely completely unusable. And we're also, ENDA is adding an enhanced filter, so you can select on certain demographic details, but that's coming up. Also, we found out on the Fast Track data page, you want to click on this little icon, hyperlink down here, not that one. So it takes you to the query tool takes you to the general query tool, and you have to then go find ABCD, but the image 3 hyperlink, it gets you straight there. So for the minimally processed data, which is with the releases, we have the SMRID, MRI and fMRI data. One of the really important aspects that sort of Don briefly mentioned is the intensity inhomogeneity correction, which improves especially occipital hyperintensities in the T1. We've seen people who've got the DICOMs from Fast Track, and then they said, well, we can't clean up the T1s because of this hyperintensity in the occipital lobe. So I'm saying minimally processed data improves a lot of the study-specific issues, especially this T1 hyperintensity. So we would really love if people use the minimally processed data when they can. The tabulated results is usually what we see at the annual release, which is all the data that we can, plus quality control metrics, and some of the metadata. Yeah. Next up is Deep, which Wes will be talking about, but the website is deep. There is the website. Genetic data, we have genetic data available. It's a smokescreen genotyping array which has got more content related to addiction, nicotine and tobacco. It's in Plink format, and yeah. So go to commonly accessed data sets and then select the genotyping data to add it to your workspace and then submit it through the filter cart. Yeah. Also, coming soon is what underpins Deep. Deep is a R-based, it's run through R, and the actual database of all the variables, we are sharing that. So if you don't want to use deep in the statistical tools in that, you can actually just download the database and then run through R your own queries. So yeah, there's a lot of data in there. I found out that we have 65,000 variables just for baseline, so that's going to increase when we have the longitudinal measures coming out in probably a year and a half. So, poll question, if everyone can get out their devices and go to GMP3 CNFIO. And we're just curious as to what type of data everyone's using. Whoa. Oh, here we go. Tabulated data, that's no surprise. We're really interested, a lot of people are using fast-track DICOMs. Cool. Oh, a lot of people are using minimally processed. That's great to see. All right, can we go to the next slide or come out of that? Yeah. Getting the hang of it now. Cool. I got the short straw to show this slide. We unfortunately we found some major errors in release 2.0, and we had to send out a notification a few days ago about this. Unfortunately, 1,100 subjects, the T1s were flipped, the left and the right were flipped the wrong way. So we are busily getting that processed, and we'll be releasing that before July 15th. Yep, I don't want to say any more about that. The next big release is the annual release 3 in summer 2020, which will have half of the second year imaging follow-up. Do know that, of course, the non-imaging follow-ups for 6 months, 1 year, are also incorporated within this. So there'll be half of the 2-year imaging follow-up, but there'll be a lot of follow-up, intermittent non-imaging follow-ups within that release. And cross-sectional measures, and then in release 4, we'll be adding in longitudinal metrics. We can't add the longitudinal metrics in at the moment because ENDA doesn't have the time to actually put it all in, because we're literally going to be adding 40,000 more metrics, I think. Associated collections and studies, for people who have gone through the ENDA collection, there's also a prediction study challenge that came out this year, and you can get the previous release, 1.1 if you want, but you need to email the help desk at ENDA. It's not generally available. The fixed release will be an associated collection, and also related publications you can find here. So as people publish with ABC data, you can see that it's associated with the collection. Also, ENDA, they have computational access through Amazon, so they have environments they can set up for you. So instead of everyone downloading all the data, they actually have the data available there. They also have a lot of free credits, and they're asking people, please ask for some, because you get a lot of them. They're kind of a bit surprised that no one's using them. So you can get a lot of credits to run your analysis, and of course, as a scalable web service, you can run a lot of processes and get stuff done quickly. So do check that out and apply for it, because there's a lot of free resources there. And also the guys are really friendly, and they'll help you set up and do the training for you, so please check it out. Associated tools, we have a GitHub page, ABCD study, where we have the code for deep. We have parsing scripts for the ePrime files, and a ability to create sifties for other software applications. Also, we have a DICOMs to bid for people using the fast track data. There's a DICOM to bids sorting tool. There's also an HCP bids service, and also an HCP pipeline, which is a modified version of the HCP pipeline with ABCD specific requirements. It doesn't run DTI at the moment, so it's mainly about fMRI. Also on Nitric, we have the pipeline as a Docker in beta. If we've got one group using it already, or yeah, but just after release 2, we've got to update that and then write some more documentation before it's available in an easier to use format. Oh yeah, here's the the HCP pipeline. And it's a bid software and works with ABCD as well as other populations. And for people familiar with Docker, that's the line to pull it down from. Also, ABCD Human Connectome project is going to be released as a collection shortly. I think it's July. So this is Damien, what's his? Damien Fair's group have been processing all this, and they're about to release it. It's in bids format, and there's an fMRI QC which is in a beta testing at the moment. Lots of people, lots of images. Yeah. Okay, recommendations. We spent a lot of time in the release notes writing recommended inclusion criteria. Of course, not everyone needs to use this, but when we've had a lot of people that say, we didn't get the results that you got, and it's like, well, if you use these recommended inclusion criteria, we can get more concordance with different studies. So it's in the release notes, in the imaging instruments release notes. And there's an example of a flowchart there. And also in DEEP, Wes is going to walk through this shortly, but you can see in select subsets for sessions, you can choose recommended T1 or T, or whichever task you're doing. And this is all for the fMRI. So extensive. There is a difference between scanner manufacturers. We can see across the grey-white contrast in the T1, mean cortical thickness is slightly lower in the Gs, and slightly different between the surface areas. And also in DTI, FA is really low on Philips, but not so much MD, and RSI neuronal, the ND measure is down again on Philips. But it's not always just the scanner manufacturers you need to control for. As you can see on the left here with mean T2 cortical grey-white matter intensity, it seems that there's a manufacturer difference, but actually per scanner instance, there is deviation, especially in the T2 per scanner instance. So this is to say that generally you need to control for manufacturer, but you may need to also control, you may instead need to control for scanner instance, which is the device serial number in the MRI info instrument, and it's also a covariate option in DEEP. So really look at your data, look at the spread of the demographics of the data per scanner instance to see whether you need to go by scanner manufacturer or by instance. It's a particular issue with T2 data. Getting help. Again, Don and I spent a very, very long months writing this very large, informative paper, and some extra additions we put in. The one that's available on bioarchives refers to 1.1, but the one that we responded to reviewers in, we added 2.0 information and a lot more baseline demographic information. We thought it would be out by now, but I don't know, it's a long document, so I suppose reviewers are taking a while to read 80 pages. Yeah. The ENDA help desk Again, I'm pointing you to the tutorials, which are really quite useful. If that fails, please email the help desk. But this is mainly about getting access to the data, about downloading packages, troubleshooting the websites, and scheduling. And if that doesn't work, contact us, ABCD Issues at UCSD, more about the contents of the files, issues about the release notes or the release in general. And data downloads from Deep as well. And also join the email list, and that's the list there, and it'll reply back to you with an email on how to join. I saw on the forum, on the Q&A already, there was a question about whether we're going to have a work group available where ABC users are able to log their questions and be able to reply to that. I'm a fan of that. Because I would like it to be something like the Free Surfer bulletin board. We don't have that set up at the moment, but we're working on the recompetition for funding for this, and I'm adding that in there as a way of users being able to interact with each other and also knowing issues or problems that have been solved before instead of repeating them over and over again. So yeah, I would like that sooner rather than later. And that's it from me. Thank you.
Hey everybody, Sean's over there, which means he's not going to stop me from saying, does anybody have any questions? Right, yeah, and I know that, and I think he's trying to field them as the talks go along, but it's just while we're figuring out this technical stuff, and I saw in the program that we were kind of officially supposed to have a 20-minute break. So if people want to ask questions or have any comments or anything, you know, or you can just get up and stretch. Yeah. Yeah. But have them reported on the website as well? Yeah, the bioarchives. Makes sense. Am I up? Okay. Okay. All right. That was your rest. So here we go again. Okay. We're going to talk a little bit about just some statistical issues. And you know, this is an interesting one because there's been a lot of there's a lot of excitement about this. Oh, great, we'll have huge statistical power, which I've mentioned earlier. And then also people very quickly say, yeah, and you're going to find a whole bunch of trivial effects, and the world is going to be glutted with yet another ABCD paper finding a statistically significant but vanishingly unimportant finding. And how do we deal with all of that? And so that's what we're going to talk about just a little bit here. And then Wes is going to take over and talk. You've seen the DEEP being mentioned. So this is this very, very exceptional data analysis portal that has been created. So what I thought I would do is first just give you just a very, very quick sense of the fMRI results. We just started looking at this now with the big data release of the full baseline data. And then talk just a little bit, let's just start the conversation. There's no definitive answers here about how we might start to best use, for example, this brain information data in this example, to study the correlates of psychopathology. And I just picked on ADHD symptoms, but this could be any phenotype that you're interested in. Doesn't really matter. And the take-home message is not profound. This is one that you've heard 100 times before, and of course you're hearing it again and again within our field. It's just the importance of reproducibility and how to build it into your analysis. I guess we feel quite strongly that with the sample size as large as ABCD, one really should build some form of replication or cross-validation into all your analyses, and this will hopefully arrest this kind of reproducibility angst that we have in our field. Okay, so this has been mentioned, the stop task, motor response inhibition, hit right, hit left, on occasion have to withhold responding with an adaptive algorithm. This is a very well-established, cognitive control task. The task activation maps look pretty good. We have ongoing discussions as to, just realized now that Anders is in the room, as to whether or not we should be thresholding these maps at all, but I did. These are just thresholded at an effect size of 0.2. And this is another issue too that we're trying to kind of grapple our heads around, moving beyond just, maps of p-values, because again, the statistical power is going to be so large. Should we be showing effect size maps, or what's the best way to even visualize our data? But you'll see we can get maps of successful inhibitions, we can get maps of failed inhibitions. And the other thing too that we're particularly, personally I'm particularly keen on, is with 12,000 subjects, we should be able to see robust activation. Right, but the critical question in ABCD is really these individual differences, is neurodevelopmental trajectories and how they might differ as a function of genetics, environment, family, who knows what. So we're very keen to see how well we can extract individual differences in our task activation maps. So this is just correlation between performance on the task, stop signal reaction time, and success. The monitoring center delay task has been mentioned. Just to flesh that out just a little bit, there are wins and losses that vary in magnitude. So again, it's just one of these standard trials where there's a cue says, say, in a few seconds there's going to be a target. If you respond fast enough, you can either win $5 or win 20 cents, or you could actually lose $5 if you don't respond fast enough, or lose 20 cents, or it's just neutral. You just try to play the game as best you can. So we have kind of magnitude effects and also kind of valence, avoiding loss as well as winning. I'm not showing the subcortical maps, but they look pretty good. We can look at the win anticipation, the loss anticipation, and then also the outcomes to winning and to losing, and also to avoiding loss, and so on and so forth. And again, all of these are available both as tabulated data, as has been described up on the as well as the raw, minimally pre-processed data, if you wish to process it yourself. And then finally, the NBAC, as was mentioned, there are different emotional stimuli in the faces. I'm blanking on what the emotions are: fear, angry, sadness, happiness. Some of those. I think 3, but I can't remember exactly which ones. My apologies. But it's also, and I will just tell you a quick aside. I don't have a picture to show you, but if we contrast all the trials with faces against all the trials with places, you do see, robust amygdala activation. So that was part of the motivation here, is to kind of at least build in some sort of an, affective probe. But the critical one, the task is primarily designed for, is to, assess working memory. So we have 0 back activation, 2 back, and obviously the difference between them. And then, the correlations with performance on the task. Here we just calculate D prime. So again, it's still early days. We're still really wrapping our heads around the data, but the activation patterns tend to be as expected, tend to be sensitive to individual differences. Okay. They also tend to be reliable. Now, this is very early days. We're just in the process of doing these analyses. What we did with the 7,000 or so subjects is we just split them into two groups, two well-matched groups, so 3,000 in each, and we created the activation maps for the two groups separately. And you can see they're very, very similar to one another. And if we actually just run the correlation, so vertex-wise correlations of group 1 against vertex-wise activations of group 2, the correlation is about 0.99. So, the activation patterns tend to be very, very reliable, at least for this contrast. This is the two-back activation. We can look at that as a function of sample size. So what if you didn't have 3,000, but you had 2,000 or 1,000 or 100, and we can plot that. So reliability in this contrast seems to be good. One of the reasons why I thought this might be of interest is that some of you out there may be thinking that you're running your own studies, and you might consider running for your particular study with your particular sample the ABCD protocol. So it might be nice to know how many participants you need to have, a good level of reliability so that you can compare against, if you will, our kind of, normative database. I don't have these curves yet for all the different tasks, all the different contrasts. I'm going to guess that this two-back is maybe as good as it gets, how good we are for some of those more subtle, let's say, contrasts from the reward task. I don't know, but we will be hopefully publishing this pretty soon, so you'll see those reliability curves as a function of sample size across the different tasks. Okay, so that's kind of a very quick overview of the task data. So what I was thinking is, okay, how does one go about, to talk through, how one run an analysis with this large data set? And a lot of people are interested in things like attention deficit. So we have the child behavior checklist. What we did is very quickly, we just scored all our participants. Just as a quick aside for those who are interested in ADHD, I think about 7 or 8% of our sample reports an ADHD diagnosis. But here we're just treating it as a dimensional variable, so including thousands of participants, parent report. If we do a vertex-wise regression, so this is stop success. This is a reasonable hypothesis. So kids who vary in their ADHD inattentiveness symptoms might show differences in brain function related to cognitive control, to inhibitory control, right? Makes sense. If you run that regression at a 0.005 uncorrected, that's the map you get. And actually, that's quite similar to an FDR corrected map. But you got lots of subjects, so you might want to push your threshold even higher, and you get that. Or you might even push it even higher again, right? And I mean, this could be your dissertation, and you might get all sorts of happy. You might say, look, we're seeing robust left inferior frontal gyrus for inhibitory control. The more your inattentiveness, the less the activity. Also true, you can see there for a little bit of left dorsolateral prefrontal cortex. Yippee. Right, analysis works. You can then try to quantify this. Let's take a region of interest analysis. So again, in the tabulated data, you don't have to go through any of the business of doing that, vertex-wise analysis. You could extract, let's say, the Destrio atlas. You might just simply run a regression. So across the 100 and some ROIs with some covariates, how much variance in inattentiveness are we explaining? And you'll see there we explain about 14% of the variance. So, not bad, right? One task in probing one system. Without the covariates, if it's just the brain data, you explain about 10% of the variance. Super. The question, the challenge, is now how reproducible are those effects, right? We know if you simply fit a regression, to some extent you're going to fit to the noise in the data. You're going to get an optimal regression from that particular analysis. To what extent are these results inflated just by the noise that are in these data, even though you're running it with 6,317 participants? So let's talk through how we might do that. Here's how we do it. There's a bunch of different ways. I'm just going to give you as part of the sort of education course, just one example of how we try to build in cross-validations into our analyses. Because replication is tough. You don't want to run ABCD, recruit 12,000 subjects, and have a reviewer say, fantastic, could you replicate that for me, please, right? But you can do it within your data. The procedure we use is we would typically first just sequester 20% of the data. Just put it over there, don't touch it. Take the 80% of the data and do what's called a k-fold cross-validation. So we might split the data into 5 folds. We'll take 4 of those folds, and we will build a regression model. And in our case, we do an elastic net regularization. So there are a couple of parameters that you want to tune to sort of penalize the complexity of the model, but also just make it as generalizable as possible. So the way we would do it is we would take 4/5 of the data, split it into 20 folds. We would build a regression model on 19 of those folds, where we just literally just like run through the different regularization parameters and then test them on that 20th fold and simply pick the model that works best. So by design, you're picking the model that generalizes best. And I'm not talking about a complicated model. This could just be a regression with just these regularization components. And then you just cycle through the data. So all the data gets treated as the test data once. So you're essentially going to build 20 separate models. And then from those, you simply pick the best. So which model trained on 19 of the folds did best on the 20th fold? Once you have that, you can now test it on that set-aside portion of the data. And how well your model does on that set-aside portion of the data is really how good your model is. How well does it perform on data that were not used in constructing the model? Does that make sense? So cross-validation. And you can just then, you cycle through those 5 folds, so you get 5 models. And then ultimately, you can test your final best model on that sequestered 20%. So it gives you a sort of external validation. It's not the ideal external validation because it's still part of the large ABCD study, but you've put it aside, it's as good as we can do with these data. The point of all of this, as just an example, is this helps us quantify how reliable our effects are. Or put another way, hopefully it helps you identify reliable effects. The point here is to build into your analysis the search for reliable, reproducible findings. Okay? All right, so let's go back to our data. Before we just ran a straight multiple regression. And with the covariates, we explained 14%. Without the covariates, we explained about 10% of the variance. If we then repeat that using this, data analytic framework, so same data, same ROIs. With covariance, that drops to 3% of the variance. Without covariance, it drops to 0% of the variance. So you can get a highly significant effect that'll pass any sort of threshold you can make up, but it won't generalize. There's a huge overfit to the noise in the data. And we think this is probably just the nature of the beast. With how we measure brains, with how we measure ADHD symptoms, the relationships are just not going to be that strong. So the challenge for us is to now extract the best measures of ADHD symptoms and the best measures from the brain to up that. But I think more modest results are just going to be more likely. It's not all bad. I've done this before. This is meant to be an inspiring talk, and it ends up just being a depressing talk. But you can use the same methodology. Now here we just flipped to, instead of looking at task activation, we looked to the cortical data. So just the cortical thickness in those brain regions. And again, now this, we report without the covariates, about 1% of the variance is explained, right? And the more we do analyses with these data, especially when we look at these sort of psychological measures, this is the magnitude of the effects that we're seeing. It's not, we explained 82% of the variance. That's not what we're getting. But again, a methodology like this, I think, is not that it's overly conservative or strict. If you take a more robust phenotype, so this is, BMI, so just body mass index, and use the exact same methodology, the structural data will explain about 20 to 25% of the variance. And again, that's 20 to 25% of the data that was sequestered over there that was not part of the model construction. So I think this is, it's not that this methodology is too conservative, is too harsh. If the effects are there, I think they're detectable. And I personally think, one could then trust that number much more than some of the, some of the inflated numbers you'll see in the literature. Then, of course, you can also, and this is the real excitement, right, with a big longitudinal study, is we can now start to see, can we do prediction? So very, very simply, instead of putting in BMI for the baseline data, as Terry mentioned in the 2.0 release, we also release as much data from the 1-year follow-up as is available up to that point. So about half or a little fewer than half of the participants, there are data available. So if we now try to do the same analysis but predict BMI a year later, in this, actually, it's not exactly BMI. It's just that we identified a group that put on a lot of weight, essentially, after the 1 year. So we flip it to a logistic regression. In the set-aside data, you get an area in the curve of about 0.77, 0.73, right? So there are effects in the data for sure, and they are detectable. We just have to, I think, be very careful that we don't start running, multiple regressions and multiple analysis and start coming up with a whole slew of what might ultimately prove to be spurious results. So essentially the take-home message here is just, again, the same. Again, nothing you haven't heard before, but just we would really encourage people to be thinking about reproducibility and reliability with this super large data set from the very get-go, from the outset. And then the last point I'll make, and this hopefully segues nicely to what Wes is going to talk about, this is going to be an interesting one. I mean, time is going to tell. Our structural data, in this first analysis explained 1%. Now, of course, if you combine that with functional data and resting state connectivity and some genetics and a whole bunch of psych assessments, I mean, this is the challenge. Can we bump that up? Can we explain more and more and more? But certainly, my sense thus far is if you're working with one modality, we should be setting our sights a little bit lower as to the magnitude of the effects that we are anticipating to see. That's meant to be inspirational. Okay, over to Wes.
Hi, so my talk's not going to focus on the small effect size so much, but I just like to comment that I don't think this is particular to ABCD. This is also something that's been seen in, say, the UK Biobank, in like the large Scandinavian population registers, that you see smaller effects than you do in highly ascertained studies, especially for neuropsychiatric outcomes. So I think it's just the nature of the beast. Yeah, I see. No, they flipped it. Sorry, it should be the next one. So, I think that's going to be the challenge going forward, is how to, I think our job as a consortium is to get as rich of a data set as possible for both environmental exposures and genetics and also for brain imaging and for neuropsychiatric outcomes to enable construction of complex models, because I don't think simple models are going to explain a lot of the variance that's, my gut feeling is like 1% variance explained is going to be a large effect. And that's kind of what's happening so far. So I'm going to talk a little bit about the study design and an informatics tool that we put together, the DAIC, for called the DEEP, or the Data Exploration Analysis Portal. I think the questions that he brought up are really interesting, but we're pressed for time, so I'm going to focus on this for my talk. So here's the just kind of some of the study aims for ABCD. So developing national standards for normal brain development in youth. That's where the population structure, the population sample of ABCD is really crucial, so that we can get like kind of a normative assessment for the population of the United States as a whole. And then, like Terry was saying, you can then, or I think he was saying at the beginning, if you can find subgroups for which there's a differential type of development, and you can assess that against the population average. And so that might be informative for psychopathology or non-normal development. We also have this trajectory perspective, and so far we have essentially the baseline data released, and that's interesting, and it's large, and there's a lot of stuff you can do with it, but there'll be a lot more you can do with the longitudinal data, with the trajectory data. And there also, I think there's a possibility that effects might be larger in trajectories. We don't know that yet. That's a supposition that people have. If you have a person who serves as their own control, or if you can look and see how trajectories differ in a population, that might give you larger effect sizes. But that's something we'll have to see. We'll have to wait and get a few more years worth of data, I think. And also examining the roles of genetics versus environmental factors on development, as well as the interaction of genetics and environment. And that also brings another point, and maybe this is the last one I'll talk about with respect to what Hugh was saying. So we know a lot of these phenotypes are heritable. Actually, I'll talk a little bit about that in the second talk after this one. And actually, Mike will talk a lot about that with the twin data. So we know the brain imaging phenotypes are highly heritable, well, highly, so like 30 to 60% is a pretty good rule of thumb for most complex human traits. Some are a bit more than that, some are a bit less than that, but there's a large genetic component to it. And so what people have found in genetics is that any one locus has a really small effect on phenotypes, but cumulatively they have a large effect. And I feel like that's where we have to move as a field in population neuroscience, any one exposure is going to have a small effect on the brain, but cumulatively, there might be a sizable variance explained if you take them into the totality into account. And so that's why I strongly believe that we're going to have to develop complex models that incorporate lots of variables while being careful about overfitting and making sure that the results are reproducible, both within the study and also across studies like the Human Connectome Project and other projects, the UK Biobank, for instance. So Sean already talked about how to access the data, so I'm not going to talk about that. Oh, just one thing, I don't know if you made it clear though, if the only thing you need to get access to the data is this is that your institution has this federal wide assurance, the FWA. So if you apply for the data, you have to sign a data use contract, and that has to be approved by your institution. And then NDA, the Dynamics Data Archive, will give you access to the data. And so the only requirement for that is your institution has this FWA. So it should be, hopefully, you can all get access to the data. Here's a little, you probably can't see the numbers. But this is a graphic of is there a laser pointer? I guess not. With the mouse? Oh, I see. Okay, so you can see this. Okay, yeah. So this is kind of the baseline. The release year is on the vertical here, and the assessments are on the horizontal. And so I just wanted to point out that we've had essentially 2 releases. Well, we had one intermediate release, the 1.1, but essentially the first release, 1, was roughly 40% of the baseline subjects, so not quite 5,000 subjects. And so the second release year has essentially the full baseline. It says 11,873, but it's actually 11,875, and then we had 3 more subjects after that. So the final one will be 11,878, unless something else happens again. And so that includes the entire full baseline, plus some of the follow-up assessments, up to the 6-month one, which is a small telephone interview, and then part of the first year assessment, so roughly 5,000 kids in the first year, and then even some of the 18-month, which is also, so the 6 month, every 6 months is kind of a telephone assessment. It's much briefer. So it's a rolling release schedule, and so the reason why I'm going over this is because I think you need to be aware of kind of the design aspects and the aspects of the release to be able to analyze the data appropriately. And part of it is just the release schedule, because like often. Always there will be kind of partial samples at some of the release times. And so, for instance, with the current one, we only have a partial sample for the year 1 follow-up. So it's kind of unfortunate because then you have to wait another release to get the full year 1 follow-up. On the other hand, you can use that as a device for replication. So that's what we did with the MICI Data Analysis Challenge. We actually used the release 1 data, and then they developed a prediction algorithm with the T1 weighted images for neurocognition, and then we validated it on the rest of the baseline data that came out with the release 2, which nobody else had access to until after the data were released. So you can actually use this as a way to replicate your findings in an objective way. And we also organized a special issue of Cortex of registered reports. And we did the same device there, where people trained the data on the data that were available, and then we validated it on the data that was held out by the study design. So here's also kind of a structure of what the release data look like. So the baseline, again, is actually 11,875, and you can see there's 65,854 measures per subject. You can see the different work groups that Terry mentioned: culture, environment, mental health, and so on and so forth. But the majority of it, well over 2/3, is the tabulated imaging data. Okay, and this doesn't include the images or the genotyping data. This is just the tabulated data that's available on ENDA as the spreadsheets. And then you can see that 6 months is much smaller, and then year 1 has basically all of the different work group assessments, but there's no imaging data. So the imaging data is every other year. So there's that baseline, and then again at the year 2 follow-up, and again at the year 4 follow-up, and so on and so forth. So right now, we basically, for imaging data, we have the baseline data released. So you don't have longitudinal imaging data yet. No, I think that's not true. That's released kind of. I take it. This is just the tabulated data, the curated annual releases. The fast track data is like released on it's like about a month behind. So yeah, that has longitudinal data. Yeah. So you can't. I'm sorry, you can't get access to longitudinal data on fast track. Yeah. So just some other aspects of the study design that I think are important to be cognizant of. So of these 11,875 subjects, there's a little over 8,000 singletons, which means there's like one person in the family who's part of the study. There's 1,600 non-twin siblings. There's roughly a little over 2,000 twin pairs. Twins, so 1,000, a little over 1,000 twin pairs. There's 30 triplets or 10 sets, which I was kind of surprised too. And actually, there's one incredible family with 5 kids that has 2 sets of twins and a singleton. And that's all, that you have to be aware that's within a 2-year span, right? So that this family has 5 kids within 2 years of age of each other. I would probably kill myself. So, so here's kind of a schematic of the nesting structure in ABCD. So you have the sites on top, and then within sites there's MRI machines. So most sites have 1, but some sites have 2 within. And so there's kind of levels of nesting, and then there's families nested within sites. So and then there's subjects nested within families. And so this is the type of structure that you should be aware of when you're doing your analyses. So to get appropriate degrees of freedom, you need to use, if you're doing traditional regression models, you probably want to use mixed effects models or generalized estimating equations or something that takes into account the nesting structure of the data. So that brings me to DEEP, which is the Data Exploration Analysis Portal. I'm just going to give a few overview slides, and if I have time, and if the Wi-Fi cooperates, I'll try to give a live demo. But so we'll see if it works. So this is a tool that the Data Analysis and Informatics Center, the DAIC of ABCD, put together to try to facilitate access to the ABCD data while also providing guidelines for best practices for use of the data. So this is just kind of an overview of the data set. So that 3.2 gigabyte spreadsheet is basically is what goes into the RDS file that Sean mentioned, which is so the way it works, I'll just describe it in words, is that so on ENDA, when you download the data, it's several hundred spreadsheets. So I guess a lot of you have been working with the data, so you have to kind of, you have to download it in several different spreadsheets and then merge them afterwards. And so what we did with DEEP is we already did that for you. So we downloaded the data. So there's nothing on DEEP that's not on NDA, but we downloaded the data, we merged them, and then we changed all the data types to the correct data types. So if it was categorical, we changed it to categorical, and if it was numerical, we changed it to numerical, and so on and so forth. We put that into an RDS file, and then we put that on DEEP. And that's also going to be available for download directly. It's not quite yet, but we convinced NDA that was something that would be good because lots of people inside the consortium were asking for it. And I think it's very helpful to just have all of that done for you. All the code that we used for merging the files and doing the data types and so on and so forth is available on ABCD GitHub. So you can do it yourself if you want to, or you can wait until, like, hopefully in a couple of weeks when NDA puts it on as a button, you can download, and you can download basically a 4 gigabyte RDS file and have everything done for you already. So here's the GitHub site. So you can see all the code for DEEP is here. This analysis NDA 17 is what the code that we used for the 1.1 release to do the merging to create the RDS file. We've updated that to the RDS file for NDA 18, which is the current release. We have a lot of other information on here. I would actually encourage people to upload code from their own papers in some ways. And so I published a paper with some people in the consortium with the neurocognition data, and I did the analyses in R, and I wrote the paper in R Markdown, and I uploaded the entire thing to this GitHub page. And so you can then, theoretically, if you have access to that RDS file, you can click a button and reproduce the entire paper, every sentence and period in the paper. So the point was I was trying to make a point that I wanted to have completely reproducible science, that somebody could literally reproduce my paper word for word, every number in the paper. Which I think is kind of a paradigm shift for large studies. This is something where everything should be completely reproducible. Not just in a sense that if you run it on a subset, it'll give you the same effects, but somebody should be able to get your code and run it and get the exact same numbers that you got for your publications. There's no reason that shouldn't be true. So here's the ABCD dictionary now, and for comparison, there's the ACP dictionary. So this is quite a bit larger. This was Hauke put that in there. So, and so I just want to, and so this is the portal. Maybe I'll try to, am I behind time? No, we can't. It's not connected? Oh, okay. Well, so I'll just have to go through it in slides then. Okay, so if you go to the NDA web page, so if you search for ABCD NDA, it'll pop you to the NDA web page, and where you basically get your get the information for permissions and where to download the data. And there's a bunch of buttons for downloading the data, but there's also a button there for deep. And you can click on it, and if you have permissions for the data, you use the same username and password to access deep. And so this I would recommend, even if you don't do your analyses in deep, but to look around and get some background information and to get a sense for the structure of the data. I think it's useful, just even if you just do it once. So one of the things that we've done is put together an exploration tool using a data ontology. And so if I was giving a live demo, I would show you this, but this is basically a tree structure based on the work groups of ABCD, so biospecimens, cultures and environment, imaging, mental and physical health, and so on and so forth. You can click on it, and it basically has all the data fields in the dictionary in an ontology, and so it makes it easy to explore, and it copies information over to the right page where there's actually a bar where you can search it kind of also for stuff using a Google-like, so you can search for something like intelligence, and even if the measure doesn't use the word intelligence in it, it'll pop up things involving neurocognition. And then it'll populate this with the data dictionary entry. You can click on it, and it gives you some data displays. So, for instance, for continuous variables, it'll give you histograms and summaries of the data, and so on and so forth. So it's an interactive tool for exploring the data set, and we found that that's important when you have 65,000 variables to have some kind of tool for exploring the data set, because it's not easy to navigate 65,000 variables. It takes even several minutes to just open it up as an Excel spreadsheet. So if you on a reasonable laptop. In Deep we have some R analyses built in. This is something that is you can actually modify and edit and download. But it gives you maybe a start for thinking about how we've thought about doing the analyses. So we've set it up using the GAM4 function in R, which is a multi-level model or a random effects model, accounting for site and nesting within twins, and you can the GAMs are useful because you can also do non-normal distributions like logistic regression or Poisson regression, and you can also do smooth effects if you want to do data-driven nonlinear effects and so on and so forth. That's all implemented in Deep in the analysis page, which is kind of showed here. So we have kind of a simple interactive environment for doing analyses and displaying results. You can go into expert mode and modify the R code, and then there's a button where you can go to click on a link, and it'll a place where you can save your models and run it again on Deep, or you could save it and download everything. You can download the code, the data that goes into the code, and all the tables and all the figures, and then you can do whatever you want to locally, including using it for publications. So I think that's it for this talk. Thanks. And now for something completely different. So I'll talk about the genetics data next. So this is a talk I'm giving for Chun Fan, who's the person in the DIC who's handling all the genetics analyses, but he couldn't make it here today. Is it? Okay. So I think a lot of people are probably interested in imaging genetics, and then Mike Neal, who's here, who's the head of the ABCD twin analysis efforts, is going to talk about the imaging genetics with the perspective of the twin data. But I'm going to talk about it in the perspective of the molecular genetic data, so the genotyping array. So we have, I think Sean mentioned this, we have the smokescreen genotyping array, which is almost 650,000 markers. It was supposed to be enhanced for smoking and addiction-related markers, but probably more important, it has general high-value markers you can use for imputation of the entire genome. So far we have 10,000 individuals successfully genotyped. There's some issues with QC with the ones that we don't have yet. So we'll get all of them. We'll get all the kids hopefully by the next data release, or at least the vast majority of them by the next data release. So hopefully we'll have almost 12,000 whole genome genotype arrays available by the next data release. But right now we have 10,000 available, so we have a pretty good, most of them available right now. And so we've done quality control and imputation using the Recopoli pipeline. The QC is going into what SNPs we're releasing. The imputation we're not releasing yet, and we have to discuss as a consortium if that's something we want to make. It'd be nice to make it available, but some of the outputs for that we will make available, I'm sure. So just a little bit of a sense for the quality of the data. So after doing the genotyping quality control, the average call rates are almost 99%. Roughly 150 individuals didn't pass QC, and they'll be regenotyped. And so after QC, we have roughly 550,000 markers on about 10,000 ABCD subjects. So, so for the imputation, again, this is not something that's available on NDA right now, but hopefully will be in the future, but we'll see. And so you can take this final list of SNPs and do an imputation, and you have to be careful because this is a highly admixed sample because it reflects the population of the United States. I'll talk a little bit more about that in a second. So after imputation, and by the way, so we did this at the DAIC, but then a couple of other groups in the genetics work group also did this similar analysis going through the QC, but independently, and they came up with very similar results. So I think we have something pretty close to what would be a final solution. So for a lenient QC set, we have almost 32 million SNPs, and then for a stringent QC set with stringent QC, we have about 11 million SNPs. Okay, so just as a summary of what I said so far, so ABCD is a multi-ethnic cohort with complex admixing. So I'll talk a little bit about that in a second. There's twin data, of course, but there's also a lot of relatives, sibs, and cousins. And that has to be taken into account in any kind of genetics analyses. We have roughly 550,000 SNPs after QC, and with a stringent threshold for imputation, that goes up to about 11 million SNPs or so. So the processing node and pipeline are available on ABCD GitHub, the same page I just showed, and the smoke screen array data in the p-link format are available on ENDA right now for the 10,000 kids. So here's what's available on ENDA. Here's a screenshot of the NDA page where you can access it, and it's in p-link format to make it as easy as possible for people to just get the data and start analyzing it right away. Okay, how much time do I have, Sean? Oh, it's up there. I'm sorry, I should look. 16 minutes. Okay, so I'm talking a little bit about what types of things you can do with the data. So there's some complexities and issues you need to be aware of because it is a population sample and there are a lot of related people in the data. So what are some things you can do with the data, with the genetics data once you get it? For genetic ancestry, that's often considered to be a confounding variable because in the United States, genetic ancestry is highly correlated with environmental variables. And so one thing that you can do is basically use the genetic ancestry factors, or the GAFs, which are essentially assessments of each subject's individual background as a basis for a covariate for adjusting for confounds. And I'll show you some results for why that's important in a second. And the crucial reason for doing that is that, again, not all genetic information is biologically related. In the United States, and in particular, the genetic ancestry is highly correlated with which environment you live in, and that can have an impact on outcomes as well. So I'll skip that slide. Another thing you can do is genome-wide association studies. It's a bit small for a genome-wide association study. Most modern GWAS have more than 10,000 subjects. But on the other hand, and I'll show you some preliminary results, that this is pretty constant with what the first release of the UK Biobank data, and they found roughly 400 significant hits across different imaging modalities after genome-wide correction for genome-wide testing. So this can be a basis for doing GWAS. Again, you have to be careful about doing GWAS with admix samples to control for genetic ancestry. One way of doing this that Chen used in particular is using mixed effects models. And then another thing which I think is going to be really quite common for use for the genetics data is to assess heritability. You can do that with molecular genetics. You can also do that with twin data, which Mike will show. But you can also do it with molecular genetic data. And then something that I think is going to be really common is to compute polygenic scores of different phenotypes and then apply them to the brain images and the other phenotypes that we have available in ABCD. And there's plenty of opportunities for novel analytical methods here too. And one thing I want to point out is that unlike most GWAS data, this is a very deeply phenotyped sample and a longitudinal sample and a population sample. And all of those aspects are really quite novel. There's other data sets out there that are larger than ABCD that also have imaging data, like UK Biobank. But there's no other data set that has those characteristics, that it's a longitudinal study on the same cohort. It's really carefully calibrated to match a population, and it's extremely deeply phenotyped. So I think those characteristics really make ABCD potentially a very interesting tool for doing imaging genetic analyses. But we'll see going forward. But and because of this, it's a fairly novel data set in that sense. There's some novel methods that will have to be developed to look at things like the genetics of trajectories, brain trajectories, and how that might be related to trajectories of neurocognition and psychopathology. So just some quick examples, and I think I'm probably going to end up early because I was planning on doing a deep demo. So you'll get a break after all. So with the multimodal imaging, we have structural, diffusion, task MRI, and resting state MRI. And with the genetics data, then this provides a pretty unprecedented opportunity for doing kind of population neuroscience imaging genetics. Here's an example of a preliminary GWAS that Chen ran. And so what he did is looked at cortical surface area associated with the genotypes. And I believe what he did is he took the Desicon atlas and he computed the association for each region in the Desicon atlas and then took the min p-value going forward. And so what he found was 3 or 4 genome-wide significant SNPs. So even given the relatively small sample size for ABCD, for a typical GWAS, we're finding 4 genome-wide significant SNPs, and they all replicate in the UK Biobank. So this is something that it seems to be giving us adequate results. You see on the upper left is a QQ plot of the p-values, and for this analysis we used a mixed effects model to control for genetic ancestry, and it seems to control the inflation due to genetic ancestry relatively well, though there's perhaps still a little bit of remaining inflation in the test statistics. Another thing you can do is look at genetic correlations. And again, I think Mike probably would have a lot to say about this with the twins, but you can also do that with the genotyping data. So you can look at 2 phenotypes and look at how if they have overlapping genetic causes via genetic correlation. You can do that with a bivariate GCTA type of analysis, or with a kind of a method of moments analysis like what Chen did here. And so here you can see basically on the left. The left side here is the phenotypic correlation. So this is, I believe this is the Desicon parcellation for area thickness and sulcal depth. And you can, of course, within each modality, you see a much higher correlation, but you still see some off block diagonal correlation. And then in the plot on the right, you have the same thing, but this is with genetic correlation using the Hassman-Elston regression. And you can see that it gives you essentially a very similar structure, but perhaps with a little bit less of this off-diagonal, smaller, like negative associations that you're seeing across the modalities. Just coming back to the population structure of ABCD, so here's kind of what it looks like. So on the right side is a plot of the first 2 principal components of genetic ancestry. And so you can see there's a lot. It's not just European. So this blob here is European. This is African, and this is East Asian, I believe. And so there's, this is because the United States is a very heterogeneous society. And then you can see all the people in the middle are the admixed folks in ABCD, and there's a lot of admixture. And you can see that on the left here as well. And so on the left, each column represents one person, and the color is essentially their genetic ancestry. And so you can see there's blue is European, so there's a good chunk of people who are of almost exclusively European genetic ancestry. But then there's a good chunk of the sample, over half, that has admixture, and some of it's extremely admixed. In fact, with the Hispanics are exceptionally admixed in the United States, and the admixture structure changes depending on if you're in the East Coast or the West Coast. In the East Coast, it tends to be more with African, admixed with African, and on the West Coast, it's more admixed with I guess Native European. So it's because there's more Mexican people of Mexican descent on the West Coast, and more people from the Caribbean on the East Coast, or from Puerto Rico. So this is something that is really, it creates a complication in any genetic analysis, but it's something that is important because that's just the nature of the United States. It's the nature of our sample, because our sample tries to mirror the United States. Another thing that you have to be careful about when you're doing genetic analyses is the kinship. And so we have, by design, we have twins. And this is pi hat is a measure of genetic relatedness, where 0 is unrelated, and 1 means you have identical genomes. So you can see, if you focus in on the 0.5 and above, we of course have a bunch of people who have identical genomes, either the monozygotic twins in ABCD, and then we have the dizygotic twins and the SIBs, and we also have cousins and half-brothers and so on and so forth. So there's a lot of complexity in the family data. And so as of now, you can capture that in the genetics, but going forward as a consortium, we're going to come up with more graded, more complex ways of capturing family structure via genetics or via living in the same household, or both, or using the pi hat assessments as a measure of relatedness and so forth. And so there's a lot of different ways you can define families. And so that's something we'll have to explore going forward in ABCD. Just as a snapshot of why it's important to control for genetic ancestry in association analyses. So this is an analysis where Chun took the GTI measures, surface area, and NIH toolbox, and used those to predict genetic ancestry. So you can see there's associations with genetic ancestry for all of these measures. And so if you don't control for that as a confounding factor, you're going to fall into some traps. So you'll have some spurious results. But here's an example where we took the polygenic risk score for educational attainment, which seems like a weird phenotype, but it happens to be one of the largest meta-analysis GWASs out there. It's got over 1 million people in it. And it's been predictive of neurocognition and other outcomes. And so in this analysis, we computed the polygenic risk score of educational attainment based on the summary statistics from the educational attainment meta-analysis GWAS, and then applied that to the ABCD subjects, and then controlling for genetic ancestry and other covariates. These are the associations with DTI, there's not much. Surface area, there's some, and then for NIH toolbox measures neurocognition, there's quite a bit more. So you can see that and this is pretty much in line with what we'd hope to see for the effect sizes. This is variance explained on the y-axis. That's pretty typical, I think. It's actually not bad at all. So and there's a lot of different, like I said, there's a lot of different things you can do for methods development or for interesting analyses. For example, you might use a genetic instrument for some phenotype like intelligence or psychopathology of some type. In this case, maybe say educational attainment, and this has some impact on a cognitive outcome, and perhaps that impact is mediated by its effect on the brain. So so you might want to look at, say, multimodal mediation of genetics on some ultimate outcome. Because we think the brain, like things like cognition happen in the brain. I think that's not a debatable point. So some of this stuff has to happen in the brain. The debatable thing is if we're capturing the right information that for these mediation analyses, is it contained in MRI measures or is it contained in something else? But presumably, some aspect of the genetics impact on, say, cognition happens via its impact on the brain. And so it'd be interesting to assess that. And so here, Chun did, and so with surface area, and so here, roughly 4% of the effects of the polygenic score for educational attainment on neurocognition is captured via its effect in surface area, and primarily on total surface area. You can also try to look and see if it's regionalized, but at least in this instance, it seems to be primarily driven by total surface area. So just as a summary then, the genetics data for ABCD, I think can be useful for to answer a lot of different questions, and is important because brain imaging and other complex human traits, neurocognition, psychopathology, we know for sure have a strong, substantial genetic component. Mike's going to show a little bit about that with the twin data, but you can also show it with the molecular genetics data. And so you can do genetics analyses on imaging phenotypes. You can use genetics, outputs from genetics data to control for potential confounding effects. And then the last one is just a comment on like looking at, say, global versus local effects in images, which I think is an interesting question to look at going forward. So thanks.
Does that work? Okay, good afternoon, everyone. My name's Mike Neal. It's been 30 years since my last conference in Rome, and it's really a great honor and privilege to be here again. I want to talk about some of the approaches to analyzing the twin data, and not just sort of how heritable stuff is, which you can get bored of fairly quickly, but also what kinds of causal inferences twin and family data can help you make. So never mind all the clicker business, but anyone familiar with structural equation modeling here? Yeah, goodly number. Okay. Bad luck for you guys, because I'm actually going to tell you a bit about how it works. And I'll talk a bit about heritability and what it really assesses, and talk about how we can deal with particularly site effects in the ABCD in the context of the twin analysis. I'll present a few of the univariate and some bivariate results. So things kind of start for a structural equation model. This kind of 3 latent variable and 1 observed variable model has been around in twin data analysis for really a very long time now, over half a century. And the essential principle in drawing out a diagram here is that the measured stuff is, we can annotate this a little bit, the measured stuff is in squares. So we measured a pair of twins here, both on the same variable. And those variables are assumed to regress linearly on an additive genetic component, which perforce correlates 1 between MZ pairs, but only 0.5 in DZ pairs. And in reality, in the ABCD data set, we can actually do a bit better than that because of the SNP chip data, we can measure the genetic correlation between the DZ pairs, and it typically varies sort of 0.42 to 0.58, something like that. In addition, we assume that some of the shared background, being in the uterus at the same time and all that sort of thing, will generate shared environment effects on pairs of twins. These are estimated with the regression coefficient C on this latent variable uppercase C. And then there's residual individual specific variance component E, which would include measurement error. The two-headed arrows here are sort of unspecified associations due to other factors not represented in the model. And the directional paths, this is simply not anything different than a beta regression coefficient. It's just a regression of an observed variable on a latent one. We do most of this model fitting and specification in some software that I've had the privilege of being able to develop and to contribute to its further development. It's an R package, open source, and the guinea pig here is a reference to Sewell Wright, who first developed path analysis about 100 years ago. This path coefficient model is only one kind of way of going about it, and actually we've tweaked the approach to estimating variance components in twin data recently for some statistical reasons. But basically the guts of it, you're like, okay, I've only got one variable and I've got three latent variables. How the hell are you going to, pick out three parameters out of what is really one kind of observed statistic? And the result is really arrived at by considering the expected covariances. So a structural equation model like this can generate you an expected covariance matrix, and for a pair of twins on one variable, that's just a 2x2 matrix. And there are three kinds of statistic that are really different in the data that's going into estimating this model. The covariance between MZ twins, the covariance between DZ twins, and the total phenotypic variance. And you could solve these equations, this is just like not even high school, probably middle school math or something like that, simply by doubling the difference between the MZ and the DZ covariance in order to estimate A squared, etc. Rather than just sort of manually use rote formulae for estimating variance components, we use maximum likelihood. It has some very nice properties. It'll get you the right answer in the long run if you collect all the data in the world, i.e. it is asymptotically unbiased. And secondly, of all of the estimators that get you the right answer in the long run, it is the most efficient. That is to say, if you had thousands of different studies and you measured the variation in the estimates across the studies, you would find that this estimator had the lowest variance. That is, it's the most accurate estimator. So that's sort of why we go through the hurdles of model fitting rather than just manually cranking the handle to solve some simultaneous equations. And it generalizes very nicely. The tweak that I mentioned for changing the analytical model for twin data came about partly, I think, through the advent of estimating variance components in different contexts, in animal studies, and more recently using genotypes measured in a sample of ostensibly unrelated persons. And the deal here is that, okay, so these equations look strikingly similar, and yes, I can go through exactly the same algebra and estimate the components again, but the real key difference here is that this model lets a variance component go negative. Why would we want variance components to go negative? I mean, it seems stupid, right? A variance is a squared quantity. Are we talking complex numbers here or something like that? No, it's not that. It's just that the model might not be right. So if you come across a variable where your DZ pairs are more correlated than your MZ pairs, that would actually pop up in the model as an additive genetic variance, this guy over here, being estimated as negative. So if you imagine under the null hypothesis, you've got a trait and it's got zero additive genetic variance, and you start to estimate that variance and do so over dozens of studies, half of them should be a little bit negative and half of them should be a little bit positive, and on average, they should recover what you put in, i.e. zero additive genetic variance. But if instead you put a boundary and say, okay, I'm not going to let those variance component go negative ever, what you get is a sort of 50-50 mixture of estimates of 0 and estimates that are positive. And that will give you an upward bias in heritability estimate. And that also applies to the shared environment, that both of these variance components are at risk of being underestimated by having a lower boundary of 0 imposed. So we sort of skip that these days. Okay, so once we've cranked the handle, you know, what is the meaning of this statistic? What is a heritability? And it's not a sort of immutable characteristic of a disorder. It's not some sort of speed of light constant that we're trying to estimate more precisely, speed of light, say, in a vacuum. But rather like the speed of light in different media, of course, the effects of genetic variants are going to have different effects depending on their environmental context. And it is really, it's a measure of a population at a particular time, and a particular measure of that, of people in that population at a particular time. And it can vary as a function of age or other environmental factors. And it might even not be a sort of biological thing at all. A lot of times people sort of pigeonhole heritability as being just biology and the environment as being just stuff outside. But in complex traits, human behavior for one, you can imagine, okay, so there's some genetic variance in how pretty a person looks. And then they go out into the world, and it's not too difficult to imagine that attractive-looking people get a better response from other human beings than someone who doesn't look as attractive. And yet, that attractiveness is probably very largely genetic, physical features. You don't have to look at very many pairs of MZ twins to start thinking, wow, how you look seems to have an awful lot to do with your genotype. And so here we might have a situation where the genotype is changing your physical features. That influences people and their responses to you, and perhaps their responses, how nice they are. People in this town are really, really nice. I think they probably don't have as much as in the way of problems with depression, but you could imagine that this extracorporeal effect of how I look on how other people treat me as having an indirect, through the environment, effect on my outcome phenotype of depression, for example. Okay. Indeed, we observe such things. So Hermine Maas and colleagues published a couple of years ago a map, effectively, of the heritability of nicotine use with age by accumulating basically a mega-analysis across many of the studies, twin studies worldwide. And interestingly, you know, at a young age, on the x-axis here, we have ages 13 to 16 up here, looking very largely shared environment in bright green and specific environment in blue, and not a great deal of effects of genotype. But as the kids age and they move away from home and they start to individuate, then we start to see more effects of genotype and rather less in the way of effects of the shared environment. And that's kind of to be expected. Those environmental experiences, like I think pretty much anything that happens to you, is going to decay over time. There aren't that many permanent scars that we carry around unless we have a permanently scarred genome, and that is continually throwing out scarred biological stuff. Might not be scarred, might be really great biological stuff on your phenotype. But that you're going to carry around with you. It's going to be a perpetual influence, and it may take a while before its influence shows up. This, of course, means that you can get rather strange things. So we've noted in the Human Brain Development Health Study, which was a nice setting the stage for the ABCD cohort, a collection of about 800 people with some 1,800 scans between them, including about 300 twin pairs. And there we observed that genetic and environmental variance at age 5 to 10 was roughly equal, so a heritability of around 50%. But after puberty, there turns out to be a bit of a leveling off of the reduction in genetic variance, a more rapid leveling off than there was for the environmental variance. So the heritability actually increases whilst the total genetic and environmental variances both decrease. Okay, so what does it look like in the ABCD? Well, we've already heard that there's literally 65,000 plus variables to be had in the data set. And we only looked at the continuous variables thus far, and looked at the baseline assessments. We're all in the 11,870 something. This time it was 75, not 78. And we fitted with a variety of starting values to see if there were any awkwardness. Occasionally you get warning messages saying that I'm not sure the models converge. Would you mind having another go, please? And so we got about 50,000 variables out. And then, of course, there's a challenge of how do we present such results. This, I think I can pretty much safely skip over fairly quickly. We've got lots of different sites. We've got roughly equal males and females. We've got different races. And 3 basic scanner types. And the representation of the twin samples, you can tell which are the twin sites here because they've got the big orange blocks, but amongst the 22 sites participating in the consortium, obviously we pulled together about 1,000 pairs of twins. So, of course, sort of the elephant in the methodological room of the ABCD study is the multi-site aspect. Without having 20-odd sites, we couldn't possibly assemble a data set of this size in a couple of years. And yet, the sites may differ. And so the site is another possible source of variation. Scanner, indeed, may be part of the picture for variation between sites, but with only 3 scanner manufacturers, we can't reliably estimate a random component associated with scanner. But we can do that with site. And so we developed a multi-level model in OpenMX for individual level scores. Down at the bottom is a person's phenotype. And they get from the family level their shared environment, a common genetic component, which might be all of it if you're an MZ pair, and variation due to whichever site you're measured at. Similar to the modification of the ACE model from path coefficient estimation to variance component estimation, we can make that transition here as well and avoid accumulating bias from having fixed lower bounds of 0 on parameters. And I'm particularly proud, coming from Richmond, Virginia, to have come up with a name for a parameter that is RVA, which is sort of the mnemonic for the town where I live. And essentially that RVA is how much I correlate with my relatives. So this is a bit like the 0.5 or 1 that would go into the regular twin analysis, but it's different for each pair. And so it indicates how much I get that is specific to me and how much I am sharing with my relative. We regressed out all the effects of age, race, and scanner just using LM in R, and used standardized variables on the way in just to make model fitting more robust. You don't really want to go through with a fine tooth comb 50,000 outputs and figure out which ones are right and wrong. And we got estimates of variance components for the pieces of the study that have been described earlier in this session. So looking at physical health, the general picture is coming out with largely a good dose of additive genetic in pink, some shared environment chunks in blue, and some random environment in yellow. That's a consistent color scheme. There's not a whole lot of effects of sight. There's maybe a little tweak here for testosterone. Not sure what that's due to. But there's generally rather negligible variance associated with sight. That is also true for childhood behavior, and it is sort of reassuring for this age group that by and large these results are consistent with results from other studies to date. The CBC has been very widely used in developmental genetic studies, particularly by groups in Vermont and Amsterdam. Cognitive ability, this is not looking quite as heritable as we're used to seeing. Normally we see, as Wes mentioned, sort of 30 to 60%. At this age, we're actually seeing quite a bit of shared environment variance component, and that's probably still consistent with the literature. Often we focus on cognitive abilities in adults, where the heritability, rather like the case for nicotine use, tends to be higher in adults. But at the moment, there's considerable evidence for shared family environment effects, but not so much for sight. It's something of a relief not to see great chunks of sight variance, because it would not necessarily be that easy to tease out whence it came. Culture and environment look fairly dramatically different. That unsurprisingly, measures of sort of family environment and neighborhood where you live and how many gunshots you hear every night and that sort of thing are very largely due to family environment, only a smidge of genetic variability. For parental monitoring there and school environment, etc., there's some creeping in of more additive genetic effects. And also some rather novel results for resilience, for which I don't believe that there's been much in the way of prior twin research. For mental health, similarly, we're seeing, okay, for the sum scores for the mental health as assessed by the parents, that's showing a big chunk of more than 50% additive genetic variation. But the youth scores are showing a sort of rather more even split between additive genetic and family environment components. And those two only sum to about a third of the variance. Residential history. Well, surprise, surprise, there's a good deal of correlation between twins. So since they're perfectly correlated on family environment and on sight, this is saying that basically the MZ and the DZ correlations for these variables are both very close to 1. Again, somewhat reassuring to see that kind of result come out. For the neuroimaging tasks, we have both the structural and mostly atlas-based. We haven't gone as far as voxel-based analyses as yet, but we can have a look at the DTI, the structural and the functional neuroimaging tasks, and peek at those. Of course, there's very large numbers of these. For structural MRI, we can see these. You probably can't see, but I can just about make it out. The y-axis here goes from 0 to 1 for the proportion of variance due to additive genetic effects. For structural MRI, again, it's not sort of above 0.5 where you might have expected it. And the shared environment component is also poking its head above 0, running at about 0.2 for most variables. Random environment variance, makes up about half of the variation on average. And side effects are mostly fairly small, but for some of the measures, they are really fairly profound. That is that we're seeing maybe as much as 20% of variance due to site, and that seems to me is likely due to differences in scanner and processing pipelines. I forget, to be honest. I can tell you afterwards. I have good news from Anders on that front. He's been working very hard on new methods of data harmonization from across different technologies and analytic pipelines. And it'll be very reassuring if we see the side effects decay as the harmonization improves. For functional MRI, kind of a big spread here, that the variance, it's not as though that we're seeing negligible heritabilities here. And indeed, some of them look like they're over 1, but that's because this isn't really a proportion measure when some of the variance components could be negative. There's rather negligible effects of the shared environment. There's a fairly decent spread of random environment variation, but not much site variance for functional MRI. Of course, the sort of reason for doing this is not just sort of where are sources of individual differences coming from, but one of the real sort of raison d'etre for the ABCD study was to try and figure out, okay, what is happening in development, what is causing what? You can observe an association between a small part of the brain and an adverse substance use or psychiatric outcome. What you don't know in that cross-sectional study, and what cross-sectional mediation a la Barron and Kenny is not going to tell you, is what is causing what. However, there are the usual ways around trying to identify causes. Experiment. This is a naturalistic study, so we're not doing that. The next best thing is to use statistical inference and designs that will permit some causal differentiation. The cross-lag panel design is one way of tackling it through longitudinal data, but it is also possible to tackle causation to some extent using a twin study. I'm going to explain a little bit about how that might work. And first of all, you need to think, okay, what am I going to do with two phenotypes with twin data? And one thing you can do is simply specify a covariance between two traits. So just as we partitioned the variation in one trait into different variance components, you can partition the covariance between two traits in exactly the same way. So we can get an idea whether things correlate for genetic or environmental reasons. But that's really only part one. And of course, we need to do that in the multi-level model context as well. And this, yeah, okay, so just a few of a bit of eye candy for bivariate analyses between picture vocabulary and cortical thickness. So we're seeing about a range of minus 0.3 to plus 0.3 for the genetic correlations across the brain. And it's somewhat reassuring to see the variance components beginning to look a bit like those in prior studies, but the estimation of genetic correlations is going to be less precise than the estimation of variance components themselves. So there's a bit more noise, especially in the prior studies, for estimating these statistics. But here's the comparison, and honing in on the the HBDHS study, Human Behavior Development and Health Study, led by Jay Geed. That picture's on the right, and it doesn't take too much imagination to say that these images are broadly pretty similar. So it's beginning to look like a replication coming out of the ABCD of prior genetic correlations between picture vocabulary and cortical thickness, which I think is a really encouraging sign, especially for myself since I've been involved in both studies, but maybe that's a confounder we should try and get rid of. The ABCD genetic correlations with CBCL scores also shows a somewhat interesting pattern, though I have no prior literature with which to compare it right now. And it seems like the externalizing genetic correlations, whilst being bigger, are rather more diffuse, and that there's more localization involved in the associations between internalizing disorders and surface area. So I mentioned about using twin data to make causal inferences about the relationships between variables, and people often have a hard time believing that this is possible at all. I'm going to show you roughly how it works, just crudely, and we're thinking that, okay, I've got myself a pair of twins here, I've measured their brains, they're the endophenotypes on the left-hand side, I've got, say, a substance use disorder on the right-hand side, and I can measure the correlation between twins for the brain, I can measure the correlation between twins for the substance use disorder, and I can also measure within individuals the correlation between their endophenotype, their brain, and their drugs. And that last correlation is enough information to estimate the regression coefficient here, B, but this model actually makes predictions. So this is supposed to be endophenotype causes substance use disorder, and the predicted covariance, the cross covariance between one twin's brain and the other twin's drug use is predicted to be the product of these two quantities, the correlation for the brain and the correlation, the twin correlation for the brain, and the substance use endophenotype correlation within persons. That prediction of that cross correlation, one variable in one twin, the other variable in the other, is different if we flip the direction of causation here. If we say that substance use causes endophenotype, then this association is expected to be B times the correlation for substance use disorder. And as long as that correlation isn't the same as the correlation for the endophenotypes, we get some leverage to infer which direction the causation may be going. And we're not sort of just going to stop there, but we can also allow hybrid models. So essentially the three parts of possible covariance and the two possible causal paths from one trait to the other, it turns out that we can estimate any three of what is now 5 possible sources for covariance at the same time. So obviously there's some assumptions required in here of, say, no prior genetic or environmental correlation. But you don't have to assume that they're all absent. So you could retain a model with genetic correlation and some mutual feedback loop correlation between the two traits. And we can test the identification of these models using a handy little function that Mike Hunter programmed in OpenMX. It's not that, structural equation modeling is the only way to do this kind of thing, but there are other approaches, and they have their pros and cons. The regression approach is attractive because it is more rapid. It is not involving, typically it's not involving parameter estimation through optimization and maximum likelihood, but rather simply solving the BLUP equations, if you like. And rather similar to the structural equation model, one approach is to look at intra-pair differences on one variable and see whether those are associated with intra-pair differences on another. This is a summary statistic approach, effectively, and it's rather important to not use just the differences, but also keep the sums in your regression equation as well. Otherwise, this approach will have less statistical power than that of, say, a structural equation model, because the difference between two twins of, say, a standard deviation contains a different amount of information if it's the twins are like -0.5 and +0.5 in z-scores than if they are +1, +2, and +3 standard deviations from the mean. Those differences are quantitatively different. We have yet to develop, and I think Wes talked about the opportunities for development of new methods, have yet to implement a multi-level causal twin model, but that is on the to-do list. And there are a few sort of recognizing that if you wanted to do pairwise causal analyses of 50,000 variables, you might be waiting around rather a long time. We've implemented some parallel processing methods for OpenMX, which, being an R package, the user interfaces and says, okay, here's my model, here's my data, right, go away and run it. But all of that stuff of like, oh, is the model making sense? Have I got all the bits and pieces I need to fit the model? All that stuff happening in R is relatively slow. And then the actual model fitting by design, we flatten the model, throw it over the fence to a C++ backend in order to get numerical optimization working quickly. And it occurred to me that if you want to run through doing thousands of bivariate analyses or something like that, it would be better not to do all of the bothering the R front end with taking a look at the script, making sure it's the right way up and all of that, but instead to just sort of automatically reload new data, fit the same model, and just pump the output straight out to a file. And this is sort of phase one, if you like, of making what I would see as becoming a kind of standalone and pipeline-friendly version of OpenMX, where you wouldn't have to bother with R at all, but you might have pre-built a script using R that it could read and thereby skip the R piece altogether. And another way around that's already had a mention because of its potential access through Deep, though I must admit I've not been successful so far in using the cloud computing resources that Deep offers. But we published a paper last year showing a replication of work done by Eric Schmidt that took like 2 months using 20 nodes on our cluster at VCU. And instead, with the help of Joshua Pratikin, pumped it through the cloud using 16,000 nodes in the space of a couple of hours. And that kind of thing is basically a game changer. And with that, and being over time, I'll thank you for your attention and happily take any questions.
So we'll do slightly unorthodox, but we're going to be asking you some questions at the beginning. And then I've been answering some of the questions online but we'll have it listed up here with the questions that are remaining to be answered, and then we'll have a chance to go around the audience with additional questions. So can everyone please make sure that their device is on this web page, and then select the afternoon course, Researcher's Guide to ABCD. Hi, Dylan. All right. So what aspect of ABCD study are you interested in investigating? And you can choose more than one. Did you guys want to see this? Terry? Oh, okay, yeah. All right. Could we have the interactive slide? Yep. Brain development. Okay, cool. Yeah, so a lot of psychiatric and neurological disorders in there. Yeah. Cool, okay, thank you for that. Can I go back to the slides? Image processing, yeah. Okay, what metrics do you use in your investigation? And you can choose multiple answers on this one. Yes, T1. Wow. Oh, sorry, that's the... Oh, yeah. Oh, behavior and cognition. So there's a lot of resting state happening. That's good. Okay, thank you. Back to the slides. This is a question from Enda about using the cloud computing service. So we're just wondering what type of environment they should set up as a standard operating environment. So if we can, yeah. Ubuntu. Okay. Oh, someone wants CentOS. Okay, cool. All right, thank you for that. What statistical approach are you using with ABCD? Also, I just wanted to point out, Deep at the moment is using the tabulated data, but we are working on vertex-wise and surface-based statistical approaches at the moment, so that's a really cool aspect that's coming out. Release 3, Anders? Yeah. Yeah. Okay. Oh, so the slide, please. Yeah. Machine learning, yeah, a lot of machine learning. Cool. All right. Thank you for that. How often do you want us to contact you? Because we've previously just sent out a blast, an email, a notification when there's a problem, but we're wondering if you wanted to be known on a more regular basis. Say quarterly or biannually, or you don't care. Okay, we'll just continue releasing it, emails as we know. A lot of the questions on the Q&A online was about the a place to ask for questions and answers that can be seen with everyone. And I think that'll be a way of getting more information out to everyone rather than just a one-way email, but actually having a conversation on a bulletin board of sorts. Cool, thank you. Yes, this is from Ender as well. There was a question online as well asking about BIDS format for the DICOMs for fast track. And Ender are interested, did you want them both as a NIFTIs, as DICOMs, or as both, or you're not sure? Someone online requested about fast track being in a BIDS format, and that's what we're working towards. So if you could answer this question, we'll have an understanding of what you would like. Oh, really? Okay. So they're mainly after just the NIFTIs with the sidecar? Or both, yeah. I'll hand that back to Ender. Thank you. I think this is the last question. If it was, yeah, if you didn't have both, would it would it be a limitation on your analysis? Dylan? Well, this is why we prefer DICOM in the beginning, because there's so much more information in a DICOM than a NIFTI, and then in NIFTI you have to have the sidecar. But everyone's moving towards BIDS format, and that's the structure it is. No limitations. Yes, not sure. Okay, so it looks like we're going to be providing both. Cool, thank you for that. And that's the end of the interaction, I think. And that's the social Q&A. Can you put that up there? Oh. Okay. Do you want this? So the first question is, have you considered creating a mailing list or forum where users of ABCD data can ask questions or interact? We do have a mailing list, which I highlighted in my section, but I am interested in a forum. Dylan is here from ENDA, who actually just...
No, not from ENDA, I'm just the NIH.
NIH, sorry. Yeah, the one thing that I think of straight away is that we can't have user IDs in a forum. So there's issues of privacy, so we have to think about that. Terry, do you?
You mean the sort of
Yeah. Is the other mic on? Or?
I think you were talking about the subject IDs in the data set, right?
Yes.
Yeah. So yes, we are not. We are trying not to use those P-goods in an open conversation. For, and you're talking about a forum where you wouldn't.
It's
have
public.
To be. It's public, right?
Yeah.
So instead of being phone users, authenticated users only. Yeah.
Yeah. Just no forum. Okay. Are they on Neurostars as well? Okay. We've only got one. Have we got more than one mic? Yeah.
Have a
Yeah. Sorry, another mic's coming out for the audience. Oh.
Yeah, they just have a regular mailing list type thing that also has a web interface. So if you don't want to subscribe, you can just search the list of the material there. So that it is a mailing list, but it also has this public forum where you can just see and search. And of course, that's the most valuable thing. So you can search other people's answers, questions and answers, so you don't have to ask the same thing again.
Okay, thank you for that. I'll chase that up with the HCP people. Don, did you want to answer this? Are the image processing scripts available for those who want to know more details?
So no, they're not. We have as a Docker, we have the full pipeline available for people who want to use that. But the scripts, the functions, it's a big kind of library of tools. And the full library of tools is not publicly available, but the full pipeline is shared as a Docker that's currently on Nitric as kind of a beta testing version.
There was another question online about the BIDS, DICOM to BIDS service as being a Docker. I think Damien would be interested in doing that, and I was talking about the image intensity correction as being a small Docker applet as well.
Yeah, in general, we have the full pipeline as a Docker, and sub parts of it, pieces of the pipeline could potentially also be made into standalone tools, but that's sort of future development.
Yeah. Is the RSI censored? It's omitted, isn't it?
Right, they're omitted. Yeah.
Yeah. I'll talk about. I discussed the Neurostar just before. Will you release the school records or any other academic assessments of the youth?
Well, we have to get it first, and that's been a little bit of a challenge. We have consented all the participants for collecting school records, but one of the challenges that we're facing is that, as you probably, if you're American, you know that the sort of American school records are highly decentralized, shall we say. There's a great deal of variability in terms of what kind of records are generated by schools, how academic progress even is measured across schools. And so we've been trying to figure out a strategy for collecting more school data in terms of academic data in a way that can be comparable across our cohort. And we have been meeting with people from the education establishment and also from people who do education research who have been thinking about how to harmonize across state tests, for example, to try to at least give us something we could bring in state tests and then harmonize them across state tests and see if we could, so we're hoping to do that, but we're finding that it's a challenging thing to do. We do have, as you noticed, that we do have a reading test from the toolbox. It's a single reading test, but it's pretty good. And we just managed to get a math test into the year 3 protocol. So we will have these two kind of, for kids, academic type skill measures available, and the reading test reading data is there now because it's part of the toolbox, and we'll have that on pretty much an annual basis, or certainly a biennial basis. So anyway, it's challenging. We will release any data we get. We release all the data we get. So whenever we get it in, when we find out how to get it into the database, we release it, as long as it's not personally identifying information, of course. But we do release. We don't hold back any data. If we bring it in, it goes out in the next release. That's the policy at ABCD.
Okay. I think this is another question for you. How do ABCD researchers plan to reconcile the child and parent report versions of the KSAD?
Well, reconcile. I don't know that we do reconcile the parent and report data. They are different. They're not always congruent. So I'm not sure. I'd be happy if whoever asked that question wanted to redirect me here, but they're independently, they're collected independently, one from the youth and one from the parent. And now I know that some people who've been looking at the mental health data have been trying to look specifically at this, trying to look at how, reduce kind of the dimensionality of the mental health data, and they've noticed that there's a discrepancy between the parent and child data in some cases, and have been trying to figure out how to model that better and how to understand it better. But I think it just is what it is. It's not always entirely congruent. But maybe the questioner had something other in mind when you say reconcile.
Questioner here.
Okay. I was wondering, for instance, if they have a anxiety diagnosis according to the parent one but not the child one, and let's say you're interested in doing categorical comparison of anxiety versus other disorders, anxiety versus controls, and then like according to one case has they have anxiety, according to the other case has they don't, if there was thinking for that sort of the greater context.
Yeah. I'm sure there's good thinking, and a lot of it's probably happening in our mental health work group, of which I am not a member, so I shouldn't speak for them. But we do have a mental health work group that are looking at that. But I think you're raising some really interesting questions, and since it's all out there, you probably have some great ideas about how to handle that too. But it's all the mental health data are there, So there's nothing else that is not that we collected that's not there. So. Creative ways of making sense of this information when it's congruent, when it's not congruent. I think we're looking forward to it.
While we've got you, Terry, have the assessments been aligned to cover all of the RDoc domains? Yes? RDoc.
Oh,
RDoc.
Okay.
Sorry, it's my Australian accent.
It's his accent. Yeah, I would say that that was certainly a goal to do that, to cover all the RDoc domains to some degree. Whether they're all covered yet is another question. I think we are still, we do have the dilemma that we do have to make this study possible, and we have to worry about the burden on the families, and so we're always having to prioritize things. And there also is kind of a developmental plan a little bit, when would be the best time to begin to get something, or when would we maybe even get a better assessment if we waited a year or two for particular kinds of things. So there is a hope that we will fill out some of that stuff over time, even though we can't do everything right away. So, but we have had the RDoc dimensions in our sites in developing it was part of our mandate from some of our sponsors, as you can imagine, to keep that in our sites, and so it's our intention to cover them all somehow or another.
Okay, I think I've got a question for Anders. Are you going to use the same scanners for 10 years? If so, how do you keep the scanner's status the same as the baseline?
No, we realized that it's not really practical or to kind of force all the sites to keep the same scanners for 10 years. So, in fact, I think a number of the sites are already planning to go on the GE sites that are on the MR750 platform now. A lot of them will be going probably to Premier. This is the next kind of their GE upgrade, if you will. And so our kind of the process that we have proposed and we will kind of put in place with this upgrade would be to have each site, so we are doing scans essentially in the same individuals before and after upgrade, so that we have
like
lab
a test
data.
retest?
Yes.
Yeah.
So the same individuals scanned essentially back to back on the basically the old and the new version, and that should, we hope, give us kind of the laboratory data, if you will, to kind of calibrate, if you will, and kind of account for any systematic differences. We try to minimize any differences between scanners kind of by design. But, you know, as you can see from the data, you know, from what was presented earlier, even with our best efforts, there is going to be residual differences, and the same holds, I'm sure, for scanner upgrades. And so I think the best we can do is to try to kind of design the experiment so that we can have confident estimates of any bias due to those changes.
Okay. This question, are the scripts for performing cross-validation available? I'm not exactly sure what that question means. Is there a person who asked that question here? Oh, sorry.
So, for Deep, we're planning on putting in a cross-validation component with this guy. Hugh had the idea of doing that a while ago, and with discussion with the biostats work group, which Mike and Tom are part of, we'll institute something on Deep that does cross-validation. But if it was a particular question about Hugh's scripts, you have to ask Hugh.
I guess kind of further to that question, I guess it's also the kind of the permutation testing and other kind of approaches that so Tom Nichols and his colleagues have been helpful in pointing us to tools that allow us to basically do kind of permutation kind of testing in the context of structured, nested designs like in ABCD, and it turns out to be important to get valid non-parametric statistics. So cross-validation, of course, is also, I think, also would have to kind of be done in a way that actually respects kind of your variance groups and things, the nesting structure of the design.
Yeah, if the question is specific about the example that we used, that was just done in my lab. But so as Wes says, there is a plan to build this into the Deep, and then that code would be available, and it will essentially achieve the same end results, right?
And again, like I'm encouraging everybody who does analyses on ABCD and you have a publication or something up on bioRxiv to make the scripts publicly available, because I think it's important to share those resources and enable people to replicate your results and to also build off whatever work that you've done.
There was a question, have you seen issues related to the differences in the scanner vendors? I did provide some information during my talk. I've actually written 3 baseline papers on structural DTI and fMRI, looking at scanner variations, head coil. There was a scanner upgrade in GE, and do these items affect the output from FreeSurfer? I'm sure some of you in this room, with the error that came through recently, I can't submit that because I'm going to wait until release 2.0.1 with the fix so I can get the whole baseline. But a lot of that information is in those baseline papers. The main thing is there is generally there's a manufacturer effect across pretty much everything. The head coil channels didn't affect the outcomes. The software upgrade didn't change the outcomes. So yeah, the main thing is to keep an eye out for scanner manufacturer, and for DTI and T2, you may want to go down to the scanner instance because they're more sensitive. With the left-right flip mentioned, we detected it. For release 2, we didn't have time to do the DMRI QC. We've got a team of really diligent SRAs that do all the visual QC of the raw and the FreeSurfer. And so to get release 2 out the door, we didn't have time for the diffusion QC. So after release 2, then they got to the diffusion, and they noticed that the diffusion wasn't overlaying on the T1 properly, and that's when we all got very sad. Yes, and we've had to pull that back. So just like yourselves, we've had to stop a lot of papers until the fix comes out. So we apologise for that. Terry. When you start to ask questions to children as they grow older that were originally designed to be answered by adults, parents of 18-year-olds might not be too informed anymore? When do you start asking questions to children, which are adult questions? When do you flip from youth to sort of adolescent questioning?
Well, I mean, this is an important question because I didn't say enough about this in the beginning, but we do have these expert work groups in each of the domains. And they're people who are very, for the most part, they're very familiar with these methods. They're familiar with the instruments that are used across the developmental age span, and they are sensitive to the fact that they've been validated for use in oftentimes they've been validated for use in only a smaller age range than what we intend to see. And so really a big part of their charge has been to make proposals for protocols that use the best instruments that are available and that are feasible for us to use, but to make modifications if necessary to address the developmental issues. So if we're trying to bring the age down a little bit from where an instrument really was developed, it's their job to try to figure out if there are a few items that need to be changed because they're just not appropriate for the age range and so on. And there are some changes like that in the protocol. This is something we've been talking to INDA about because we may have an instrument where a few items have been removed because they're just not appropriate for 9-year-olds because it was really a 12 or 13-year-old instrument, and we've tried to bring it down and so on. So there are these changes, and I think they've all been noted in the publications and in the notes associated in the data dictionaries and so on. But there is part of what the work groups do, which is specifically to look at whether something is developmentally appropriate and to change things and make them more developmentally appropriate for each subsequent visit. So there's a fairly long process that the ABCD work groups go through in proposing their 2-year, they're always 2 years ahead at sort of trying to modify the protocol to make sure that it is appropriate for kids of that age. They're adding adult questions, I think was part of the question here, those kind of more adult assessments as they become appropriate. And it's tricky because obviously we're trying to, have a longitudinal study where we're measuring the same constructs over time, but you simply can't avoid this. I mean, there's a lot of difference in, between a 9-year-old and a 20-year-old, and you have to, kind of do the best you can. So it's an important focus. It was a complicated answer because it's thought through by the work groups, and then the proposals are brought forth, and then all the work groups talk together about it, and it's just one of those issues that they deal with.
Okay.
We might just add as well that before any new assessment protocol is rolled out, we pilot it. So we have kind of a cohort of kids who are not part of our study who, do every, the upcoming battery in advance. So if they're having any difficulty understanding a question, if there's some problems, hopefully we'll catch that before then that annual assessment comes around.
There's a question, will the slides from today be available? Yes, they should be available for download from OHBM. In previous conferences, I've found they're quite late, but they do come up maybe next week. So there's that. Terry, why does the KSAD skip neurodevelopmental disorders and many anxiety disorders that occur early on? The KSAD skips neurodevelopmental disorders and anxiety disorders, which would be appropriate for this age group.
Oh, skip some of the modules, you mean?
No, the neurodevelopmental disorders.
The neurodevelopmental disorders.
And the anxiety disorders. Yeah.
Well, I'm not sure what the question is. The KSADS has a set of modules, and I think they were all listed on one of those slides there.
Okay.
Those are the modules that are a part of the KSADS. And the KSADS, if the question is why are there things that are not there, you'd have to ask the developers about that of the KSADS. If you're saying why are we not giving them, and I'm not quite sure I understood the question that way.
I think it's a general question about why does the KSAD not, why does
it not have, the KSADS itself is a structured psychiatric interview that we did not develop, that was adopted by ABCD for looking at the sort of diagnostic level psychiatric information that we were going to be collecting. So it is designed to do diagnoses, and those are the diagnoses. Those modules are the ones that are in the KSADS.
Okay. I was going to...
But I think it was related to
Oh, youth versus parent.
Oh, it's okay. I'm just going to open it up to the floor for questions.
Yeah, just a quick question, actually. I was also wondering, as a related note, are the tasks, the fMRI tasks that were done, also potentially available? Because I think it would be really interesting on new patient cohorts that we want to run to actually be able to use the same exact task and then be able to compare to this valuable resource.
Yeah, the stimulus files, for the E-prime stimulus files, they're supposed to be on the ABCD website for download through ENDA, I think. We talked about it. If it's not there, it will be soon.
Hi, a question about the twin models. The direction of causality modeling is notoriously power hungry. And have you conducted power analysis on what kind of associations we can do actually causality model on? And again, lots of phenotypes are not very heritable. Is this an issue?
They're not necessarily that power hungry, actually. That's a slight sort of overstatement. They get power hungry when those correlations for MZ correlations are not the same. If they're exactly the same for the two traits and the DZ correlations are the same for the two traits, then you've got no power whatsoever. So it's mostly just a kind of effect size deal. There's a couple of things that help us out somewhat, which is first, in those analyses that I presented and that I forgot to mention, we actually throw in everyone who's not a twin as well. They just sort of go in without a co-twin, but for multivariate analyses, that's very helpful because it gives us a really precise fix on the within-person correlations. And therefore, we have very large sample size for those statistics, but obviously the twin correlation statistics are based on smaller sample sizes.
So what's the typical correlation that we find in a duplicate that we then hope to do direction of causality for the analysis of?
You mean a sort of rule of thumb about how different the twin correlations need to be? Actually, I don't have a good sense of that. Mainly because, well, with 50,000 variables, I think we're going to find things all over the map in terms of how much power you have. You know, if you go around selecting variables with very different familial correlations, you'll probably be in great shape. But for your particular variables of interest, that's going to be more down to chance. I would say that I think these things will develop differently. So there may not be substantial differences between pairs of variables of interest now, but in future waves, one might expect them to emerge, just as we see the genetic architecture of different traits is more or less variable over time.
I actually have a two-part question, but so have you noticed with the use of perspective motion correction for the Siemens kids who were scanned with the VNav system, that their data quality of their T1s and T2s are better than some of the other scanners? So that's my first question. And then I haven't had an opportunity to look at the 2-year follow-up from baseline, but is there any interest or including other types of perspective motion correction for the other types of scanners as new technology obviously has come out?
So I believe all of the Siemens and the NGE scanner sites use perspective motion correction. So on the Siemens scanners, it is VNavs, as you say, from Mass General. And we're working with Siemens, kind of pushing them to actually support it kind of more directly as a kind of a whip or in product, ideally. GE already has it in product, as you know, what we call promo. It's got some that's got somewhat better performance in terms because it uses a spiral navigator, so it's not kind of confounded by B0 drifts and things like the VNavs. But in both cases, I think it's very clear that the advantage of using prospective motion correction is pretty is for kids that move. It's a huge issue, and we have a number of papers on that. Philips is the one vendor. We only have 3 Philips sites, but Philips does not support any prospective motion correction currently. So right now we don't have really within the vendor in ABCD kind of with and without kind of comparison, unfortunately, in a way. So the comparison would have to be between GE and Siemens and a few Philips sites. I don't think we want to change kind of the technologies going forward on the Siemens and GE scanners if you can avoid it, again, for consistency, but we would love to kind of try to push Philips to actually have some solution. It doesn't have to be our solution, but at least a solution. I don't know if that answers your question.
Yeah, I guess for like expanding on the Siemens though, like I didn't look into the DTI data set, but like, DVNav is now, like as far as using the VNav system with diffusion data, like I don't know if that is currently being implemented or like if you've seen any.
So I think that's, for me, it's much less of an issue for diffusion and say fMRI where you have essentially a single shot kind of imaging. Then, we have some methods that for improved retrospective kind of correction, which works well for kind of for multi-band acquisitions for the EPI. So I don't think that prospective motion correction is really all that important when it comes to kind of EPI acquisitions. It's really for us. It's kind of it's the 3D structural data that's acquired over minutes, where basically there's no good way after the fact to correct for the motion. So I don't see kind of putting prospective motion correction into the diffusion or functional MRI just because it's going to have to be then change the rest of the parameters, and it could interfere. It could jeopardize or kind of the longitudinal kind of comparability of the data. So I think we're kind of locked in to some extent to technology. It would take a lot to kind of to change acquisition strategies, I would say, in the longitudinal design like this.
And with that, I think we've finished time for this session. So Terry, do you want to close it out?
Well, just thank everybody for coming, and thanks for answering all the questions. And I'm sure if you have other thoughts, we'd be happy to hear. I'd be very happy to hear more feedback from you. And if these questions stimulated ideas that you'd like for us to know or for ENDA to know, be sure and send them along, because it's all about you. It's all about the user community here. So that's
thank you very much.
Goal.